Let's start. Okay, welcome to the 7th International Workshop on Genome Privacy and Security. During this extraordinary time and in this unusual manner, right? You know, this is my first time that I have to organize an international event from my home. And I guess this could also be a very interesting experience for many of you guys. Due to the pandemic, that you know, we have to do this online. And also due to the pandemic, we have to come up with a whole program in just 1.5 months, and a lot of pressure. But I think I strongly believe that the pandemic by no means undermined the importance of the things we are doing. On the contrary, I strongly believe that the ongoing pandemic actually make biomedical privacy research more important than ever. Just think about in a world we can freely exchange our information, right? And we can share all kinds of data. We can pull the best out of mankind to come up with the treatment therapy and come up with vaccine. The world would become a much better place. But in the meantime, we still need to take care of our patients. We need to protect their privacy. We need to address, unfortunately, some man-made barrier. I think a biomedical privacy technique will get us there, will help us address those challenges to overcome the barrier, and more importantly, better prepare us for the next public health crisis. I think I'm not the only person who believes that, because a lot of people are supporting us. So that's the reason we could come up with a wonderful program and a fully packed 3 hours. So today's program will start with Dr. Mark Gerstein's and the keynote, and then followed by 2 sessions, one on privacy preserving sharing of biomedical data, while the other on the confidential computing on biomedical data analysis. And finally, we'll have a wonderful session, wonderful panel session, and organized by colleagues from Vanderbilt. They will share with us their experience on biomedical privacy research. During the workshop, I would like to suggest that we follow this protocol. During the question and answering session, whenever you have a question, you can always click this button to raise your hand. And the session chair will turn on your microphone and let you interact with the speaker. But please keep in mind, we only have 3 hours where we have to strictly follow schedule. So if your question cannot be answered, please use this chat box, and we will try our best to answer your question offline. And before we can start the program, I would like to show my gratitude to a lot of people supporting us. The first is the program committee, and we spent a lot of time carefully review all the submissions we received. And each submission got 3 reviews, and we have extensive discussion, and some submission get rejected, although they got a very good score because and only have 3 hours, unfortunately. I hope next time we'll have a longer time. And I also would like to thank GA4GH for the logistics support and the steering committee for the guidance and advice, and the student volunteer for their support in building up the website, building up the conference systems, and helping promote the workshop. So last but not least, I would like to thank you, and thanks all the attendants. And without your involvement, the workshop would become meaningless, right? Without your support, and I don't think there's any chance we can make any progress on the science of biomedical privacy research, and which becomes so important in this chaotic world, unfortunately, we have to face. Thank you, and enjoy the workshop. Now I hand over the podium to Dr. Haixu Tang, and who will be the chair for Dr. Mark Gerstein's keynote. Thank you.
Okay, it's my great pleasure. And first, welcome to the workshop. And it's my great pleasure to introduce Professor Mark Gerstein to give a keynote speech for the workshop. So Mark is currently Williams Professor of Biomedical Informatics and also Professor in Biophysics and Biochemistry at Yale University. Mark has made numerous contributions to computational biology and functional genomics and system biology. He recently interested in the genome privacy issues and has introduced the first format to study the privacy issues in sharing functional genomics data, in particular gene expression data. And he and his colleagues introduced the first format for privacy preserving sharing of functional genomic data derived from RNA-seq experiments, for example. And he also contributed to the first approach, one of the first approaches to using blockchain technology for confidential and trustworthy sharing of genomic data. So without further delay, I will turn to Mark for his presentation. Mark, could you please share your screen?
Oh, okay. I'll start now. So I'll just begin. So I'm going to talk to you today about privacy and functional genomics data.
Sorry, Mark, you haven't shared your screen yet.
Oh, sorry. Oh dear. Oh no. I pressed a button, but not the right button, or not the only button there. Hold on. I think I got it. Now I got it. There it goes. Did it share? Did it share?
Thank you. Yes. Great. Thank you.
So I'm going to talk to you today about privacy and functional genomics data. Now, just a little bit of introduction on genomic privacy. You probably all know this, but you know, there's sort of a debate: is genomic privacy like the general topic of digital privacy that people talk about all the time? And obviously, every day when we're going around the internet, we're thinking a lot about the dilemmas of privacy, you know, in relation to our web searching, our email box, and so forth. And we all know that it's a big dilemma because a lot of the strength of a lot of these big data technologies really leverage aggregating lots of people's information, for instance, web search, but they're also impinge on our personal privacy. So that's, on one hand, it's sort of like that. But on the other hand, there's this notion that genomic privacy is very different. The genome is, in some sense, very more fundamental data, you know, because it's obviously embedded in every single cell in your body. You can't erase your genome. You can get a new credit card, but you can't get a new genome, and so forth. What's, I think, particularly important for privacy is that you, of course, pass your genome on to your children. So, you know, if your genome is stolen, you know, and it sits on the web, it will remain there for many years and potentially impinge on your children's privacy. And what's also important is that we don't know very much about the genome now, or how to mine things out from it, but will we know a lot in 20 years, 50 years, and so forth? We don't know. And then a final thing to just point out is that genetics has had a long history, and some of it is frankly very ethically challenged. I won't go through the details of this, but I just have a picture here of Henrietta Lacks and the, you know, large amounts of controversy associated with that particular issue. Now, so that sort of maybe give you some pause and say, oh, we don't really want to share genetic or genomic data. But I think one thing to realize is that sharing is one of the clearest mechanisms to speeding biomedical research. You know, it's just a very straightforward way of doing better large-scale mining of information. And in particular, if we share and aggregate a lot of information, we get more power. And what I mean by power here is really statistical power. You can just clearly mathematically see that if you have, you know, 1 million genomes versus 10,000 versus 100, you're going to get much more statistical power to find important disease associations. And also, even if we can share things privately, as we all know, privacy is often very cumbersome, and this is particularly the case for very large-scale data files that we tend to have in genomics. So the dilemma that we tend to face is that it's, you know, sharing large amounts of this information is good for society. There's a collective good from it. On the other hand, there's the sense that the individual can be harmed, and we have to kind of balance this. And one way of achieving this balance, I think, is to quantify the risks and the benefits and then balance them. So I gave you a little introduction to genome privacy here. Now I'm going to tell you about functional genomics data and how it's a little different from maybe the bulk of genomic privacy and so forth, has this two-sided nature. And then I'm going to talk about the leakages from this data. I'm going to focus on the main source of leakage, that's the leakage in the reads, and how we can measure it, and then also how we can plug this leakage using a particular file format we call the PBAM format. And then if I have time, I'm going to talk about much smaller but much more subtle leakages that don't directly come from the reads, but they come from the derived data, for instance, from eQTLs and from signal profiles, and how even these leak a small amount of genomic information. Okay, so now I'm going to tell you a little bit about functional genomics data. So first of all, many of the people at this meeting are very accustomed to genomic privacy, and that's sort of DNA privacy, you know, the privacy of variants. And that's what we mostly talk about. But functional genomics data is a little different. And the way I think of it is kind of has this two-sided nature. So on one hand, as I'll describe to you, functional genomics data, because it comes from the sequencing machine, has variants in it. But the variants aren't what we're actually interested in. Whereas when we're doing, you know, sort of a DNA analysis, we're actually looking at the variants. We're trying to associate them with traits and so forth. In functional genomics, often what we're trying to do is determine general overall aspects of a condition. For instance, what your gene expression is in your developmental stage, what gene expression is a particular cell type, what levels of TF chip binding are associated with disease, and so forth. And so there's this notion of this general conclusions that can potentially be abstracted away from the genotypes, even though the genotypes are very much associated with the reads that you get. Now, some of the calculations, I should point out, do use the genotypes and the reads, but the bulk of the calculations in functional genomics don't. So another aspect of functional genomics that's important to realize is that you know, our DNA variants are essentially the same in every single cell in our body. Okay, so if we sequence our genome once, for instance, from the blood, we're pretty much done, and we don't need to keep sequencing it in other cells. Now, of course, there's modifications to this for tumors and things like that, but by and large, our DNA variants are the same. That's not at all the case for functional genomics data. Now, the levels of gene expression or the levels of transcription factor binding differ amongst all our different cells, amongst all our different tissues, so you can measure them many, many, many times. And on top of that, they differ in environmental condition, in age, and so one can see this just gargantuan amount of experiments that can be done, much more so than DNA experiments. And so the question is, you know, what are we going to do to deal with this very large amount of data, which is potentially private? And this is a picture from the ENCODE project, or the project I'm involved in, which is, in this particular picture just shows for 4 individuals that have 4 different genotypes, you know, all the different experiments that are done on them. And there's many, many more experiments than, you know, just these 4 genome sequencings. Now, another important aspect is a lot of times this functional genomics data is very much tied to sensitive phenotypes. You know, people want to know what genes go up and down, in particular, say, mental disease phenotypes such as schizophrenia, bipolar, and so forth. Now, this is true also in DNA variants, but even more so in functional genomics. And so it's the phenotypes are often the really sensitive thing, you know, that someone's been phenotyped to have this condition or, you know, have this particular attribute. So I'm going to be talking to you a lot about functional genomics data today, and I thought I would just introduce a lot of the data sets that I'm going to use. Now, I'm going to profit very much from fairly open data sets that have been developed by the 1,000 Genomes Project, where they've genotyped people, and the GiaVadi's project, where they've done RNA sequencing on some of those individuals, and then the ENCODE project, where they've done other types of functional assays, such as ChIP-seq or Hi-C, on a number of individuals. Okay, and so I'm going to talk about these data sets. Now, I want to give you a little bit of sense of what functional genomics data looks like, because we're going to be drilling into it in some detail. So the beginning, of course, you get reads from the sequencing machine. You know, they're just sequences of letters and so forth. And that's, of course, the big data, right? And then people map this to the genome. And they get what's called a BAM file. I know this is the jargon that they use, of BAM. And then what they often do is they take this BAM So now we have reads that are piled up on the genome, and they reduce it to what's called a signal track, a bigwig file. And that is essentially the depth of the piling up of the reads at any given base. And so you can see here's the depth here, okay? And then if you look at a longer stretch of the genome, it might look like this, right? And then they further reduce this data. For instance, they might find peaks in it or average the level of signal over a region to get you the gene expression level of a gene or the sort of binding level at a peak and so forth. And then it would give you, say, your gene or transcript expression matrix. And of course, these are much more highly reduced amounts of data too, as we summarize it. Now, when we think about the amount of leaking information or the potential amount of leaking information, obviously the amount of variant data is largest at the very beginning in the reads and the FASTQ and the BAM, right? And now I'll show you later on, there is potentially leaking information in, or leaking variant information in some of the more derived things, you know, the peak calls, the gene expression levels, the signal tracks and so forth, but it tends to go down as we go up that stack as it makes sense. So now, one of the big sort of privacy issues in functional genomics data is the BAM files, the sort of raw files. As I described, they contain SNPs. You know, they'll be mapped to the reference genome, and you'll see the differences relative to the reference genome. And then traditionally, since these contain lots of variants in them, they're protected. You know, they're stored in things like dbGaP or EGA and so forth. And, you know, this, of course, protects them from potential adversary, but also from scientists wanting to do useful research on it. So as I'll describe to you, of course, what we want to do is we want to come up with some system that protects the data from the adversary, but allows the scientist unfettered access. And this is, of course, becoming more and more important, as I've described to you, because the amount of data, as we know, is all going up. But what's I think particularly interesting is the amount of private data is going up much more than the amount of publicly available data. And that's why it's very important to think about ways of sharing this information. Okay, so I just introduced the functional genomics data. So now I'm going to talk to you a little bit about the core topic, you know, what we can do to kind of share this information easily. So I'm going to describe what's called a linking attack, an attack that someone can make on the functional genomics data. And then I'm going to talk about how we can study this attack and reduce the amount of information leaked. So imagine we have a large amount of functional genomics data sets. You can think of the RNA-seq from, say, TCGA or GTEx or so forth. We have a lot amount of data, and the data is tagged with a phenotype. You know, maybe you know, you have cancer, you don't. This actually says you have bipolar disorder or not, and so forth. Now, what we can imagine doing is from the raw reads, we can do a yucky, messy genotyping. We can run them through a genotyping program to find the variants, okay? But it's not going to be as good as genome sequencing, but they're going to be noisy variants. And then what we can do is we can imagine from a particular person, we steal their genotype. You know, we get it from a database, or as I'll describe, we get it off a coffee cup. And then we essentially can use that genotype as kind of a key to look them up in this bigger database. Now, the genotype isn't the thing we're trying to protect here. We've stolen that. We have it. We're using it as a key. And once we have it, we can look them up in this database. And we can find out their sensitive information. You know, do they have cancer or do they have bipolar disorder? How do we do that? Essentially, we just look at the overlapping SNPs. And, you know, we can be a little bit more sophisticated about this. We can weight the SNPs by their rarity. So more rare SNPs, you know, matching them is counts for more and so forth. And then what we can do is we can look at the, that gives us a score, and then we can look at our best score versus our second best score. We match against the database, and that gives us a kind of gap. And then we can look at that gap against statistical expectation from a shuffled database. And that gives us how significant a match of this thing to the database is, okay? And then what we can also do is we can kind of look and we can see we can get very significant gaps, and we can find matches of a query individual depending on their ethnicity against, for instance, the Giavotti's RNA-seq database. And then we can progressively introduce corruption into that database by adding in false positives and watch the signal degrade. Okay, so that's a sort of standard attack scenario. Now, this type of attack, I think it's important to realize we're starting off with, you might say, sort of noisy information from the RNA sequencing, but we have perfect information on the query, the person that we're attacking. We could imagine another situation where we have perfect genomic information, but we start off with noisy or query information. We could have perfect information. And to make it even harder, we could have noisy information on both parts. Now, as we introduce more and more noise, what was a very simple lookup becomes much more complicated and becomes a statistically challenged lookup that we have to use more advanced techniques, and we have to do a bit more carefully, particularly we have noisy data on both sides. And I'm going to talk about that in a second. So let's talk about that challenge. So now imagine we have an RNA-seq database that we're going to genotype as we did before, but now, as opposed to having a perfect stolen genotype, we're going to get some SNPs off of glass. This is the sort of paradigm more of a coffee cup here. And we're going to take, these are very noisy SNPs, and we're going to try to take them and link them. And so now what we can ask is, how well do we do the linking versus this random expectation? And it turns out we can do pretty well. We can do well with 10x sequencing on the SNPs. And for fun, what we can do is we can downsample that sequencing and say, oh, how well do we do with 5x, all the way down to, say, about 1x sequencing. And the less sequencing we do, that actually just directly translates into, it costs less. So it might cost about $1,000 to sequence the whole genome from a coffee cup, to 10x. But actually, if we're willing to go down to 0.1x or so, it only costs about $20. And so it's very cheap. But what's interesting here is we can still do the linking down at these very low coverages, because we get a few SNPs, even though they're very noisy, there's still enough to do the linking. Now I'll introduce another technical thing I'm going to come into. I kind of described to you before this notion of downsampling. So we can downsample the reads on the coffee cup, of course, and we can downsample the reads also on the functional genomics data and say, well, how well do we do when we downsample them and so forth. And another important aspect is imputation. Many of you know that the SNPs in our genome are all correlated with each other. So often if we get one, we can statistically infer the other. So we can kind of bulk up our SNP set by imputing it. And I won't go through the details of this slide, and I won't go through the slide in great detail, but I'll just say that one of the things we find is that obviously imputation increases your accuracy of linking. But if you do it on the RNA-seq database, but actually on the coffee cup, it doesn't actually help you that much. It actually hinders you. And I think it's kind of a sort of like noise, compounding noise issue and so forth. Now, another thing we can do, which is kind of fun, is I've talked to you about looking at an RNA-seq database, which is our main sort of databases. But as you probably know, there's many different types of functional genomics data. And so we can actually ask, oh, well, how do we do with all different assays? And so here's a picture, for instance, that shows versus as we sample more and more coverage, how we do with many different assays. So here are many assays. Here are WGS-like assays. Haixu is kind of like that. We can do RNA-seq-like assays and so forth. There's many different types of them, actually, including single-cell RNA-seq. We can do TF-chip assays, histone modification assays. What do we see here, which is kind of interesting? Well, what's interesting is at very low coverages down here, we actually do better with TF-chip than even whole genome sequencing, because you're really sequencing very deeply at particular spots. So you do get a few accurate SNPs, whereas WGS is all aggregated over the genome. Obviously, with higher coverages, you always do better with WGS, but Haixu essentially does as well as whole genome sequencing, as people kind of know. And you can also see here the single cell, even though it's very grotty, you do get appreciable identifiable information from it. Okay, so that's the leakage from the reads in functional genomics. Now, what are we going to do about it? Can we, in addition to measuring it, can we kind of stop the flow? So we introduced this notion of kind of a kind of privacy-preserving transformation on the data, that we remove a very minimal number of variants and try to see if we can essentially prevent this leakage of information in these linking attacks. And the idea is shown here. So we have a BAM file. And then we have something we do to it. Now, let's just, for simplicity, think of it as maybe we compute a signal profile, we could compute a gene expression level, we can compute peaks, but just for simplicity, we get a signal, a gene, sort of a signal profile across the genome. Now, what we can also do is we can take that BAM file and we can apply this transformation, PQR, okay? And what PQR is going to do is going to take away a certain number of variants, which related to this threshold R, to create a new BAM file, B star, and then we're going to do the same thing to that file, for instance, get a signal track, okay? And then, of course, we have two signal tracks here. We can compare them. We can get a difference, and we can measure the absolute value of the difference with this E sub I at each base here, okay? Now, from that, we can make some very simple definitions here. So we can define something as kind of delta private in relation to the fraction of variants that we've masked. You know, obviously, we can mask all the variants. We can take all the variants away, or we can take 5% away, and so forth. And so that's our privacy parameter. We can also come up with, most importantly, a utility parameter. And that is how many of these bases in the signal track are significantly perturbed when you mask. So for instance, you might only find 5% of them are perturbed, giving you 95% what we call epsilon utility, or maybe 10% of them are perturbed. Now you might say, well, what does perturbed mean? We have to use a threshold gamma for that. And that gamma could either be set at 0, where there's any perturbation is seen as important, or what I think is most useful is to think of that perturbation in terms of replicates. Imagine we did 2 replicates of an experiment. That would give you kind of the natural level of perturbation that you would see. And if you set your threshold there, then your privacy preserved thing would be essentially this equivalent to another replicate. So we've developed some software to do this transformation. The software takes a FASTQ file. We have it sort of shown with a lock here because it's full of kind of radioactive private data. And then we make a BAM from it. And then what we can do with our software, we call P-tools, we can make what we call PBAM, where we've kind of removed or sanitized a lot of the variants from it. And we have a diff file that we can combine with that file again, which has it's the highly radioactive plutonium now, the most private bits that we've taken off that file. But it's now a very small file, much more easy to transport, to share appropriately and so forth. And then we have our PBAM, which we have as green here. And we can do the same thing for CRAM file format as well. So now we can, with our privacy parameter, delta, the amount of variants we've removed, and our utility parameter, we can, for a simple thing like a signal track and the sort of known distribution of SIPs, we can get a kind of upper bound as we introduce, indels and deletion, or indels into the, in the variants, or what type of leakage do we expect? And we can get this kind of upper bound relationship. But this is just a bound, and probably more useful is to actually look at these files. So let me show you what actually happens here. So if you have the reference genome, if you have a BAM read that has a variant, and now you're going to mask that, you're going to essentially put it back in the PBAM selectively. Okay, so now what happens? So let's look here. Here we have, this is an RNA-seq file from this individual called NA12878. We have 2 replicates. Here's the signal file going up and down, bouncing around. And then we have our difference in the signal of the PBAM versus the BAM file. And you can see there's some difference here, okay, as we've masked things out. Now I'm going to zoom into that region with the difference. And so now I'm going to show you the 2 replicates in the real BAM and the 2 replicas of the PBAM. You can see it's very essentially indistinguishable. And the difference between the privatized BAM versus the replicate difference is essentially 0. And this is, of course, for looking at signal tracks. We can do the same thing for looking at gene expression levels. We can plot one gene expression level on a replicate versus another. That's shown in the red dots. And we can do the same thing for the PBAM. And so you can kind of get this practical where the privatized file is essentially the same as one of the replicates. Now you might ask, well, is the privatized file, can we protect it from linking attacks? And that's, of course, the key thing we want to do. So now what we're going to do is we're going to take the NA12878 file, and we're going to remove variants from it. So we're going to increase the privacy, increase the delta parameter, right, to 100% privacy, okay? And first of all, as we do that, we're obviously removing SNPs. And here we show the number of GWAS SNPs, so that's kind of a phenotype-associated SNPs removed, and they get removed, okay? But what's most important is as we remove things, we can ask what gap value do we get in linking? And that gap value goes down, it goes down. Notice that around here, around 80%, it's almost down at 0, okay? So we've there's no more possibility of doing this linking attack, which means all effective information has been removed. Well, there's still a few variants in, but they're not useful for linking. And then we can ask, how have we degraded the utility of the file? Well, obviously up here at 100%, we have degraded the utility of the file, and that's shown by this, the average error introduced. This is our epsilon parameter. But you can see around 80%, we kind of have a kind of Goldilocks place where we don't degrade the utility that much, but we've essentially sanitized the file. Okay, so that's our PBAM format. And now I'll just quickly say a little bit about this ancillary tool we've developed called FANCY. Now, these are big calculations. To genotype and RNA-seq is a lot expensive to do that, takes a lot of time to do that, complicated calculations. But it turns out you can kind of estimate the number of variants leaking by just looking at the statistical properties of this thing. Well, what's the coverage of the experiment? How much does the experiment cover of the genome? When you look at the distribution of the depth in the genome, how is it skewed and so forth, and we take all these parameters of the functional genomics experiment, we put into a little machine learning tool we call FANCY, and then we can kind of predict the number of leaking variants. And you can do pretty well in your prediction. This is just useful for someone trying to estimate how private their experiment is kind of ahead of time. Okay. So now I've talked to you about the functional genomics data and the main source of leakage in the data, the reads, how we can kind of sanitize them a little bit with our PBAM format. So now what I'm going to do is I'm going to shift gears a little bit. I'm going to say, well, even if we've sanitized most of the information from the reads, there's still subtle sources of leakage. Now, these are subtle. They're much lower number, but they're still sources of leakage. And I'm going to talk to you about two sources of leakage. They're much more subtle: EQTL leakage and signal profile leakage. So EQTL leakage is a little bit more complicated to see. So basically what happens in EQTL is a relationship between the expression level of a gene and a nearby variant, where, for instance, if you have a particular genotype, you'll, for instance, here's a CC, you'll tend to have higher expression level at a particular gene than if you have another one, say TT, and maybe CT is in the middle and so forth. And people do, for instance, GTEx is very well known for doing a tremendous number of these correlations, correlating the genotype of nearby gene or nearby variants with the expression level of genes to find these EQTLs. And this shows you cryptically how variants are associated with gene expression. Okay, so now what we're going to do is kind of invert this. And first we have to make some definitions. And so we're going to make a definition, first of all, how predictable an EQTL is of genotype. And this is very much related to how correlated the genotype is to the gene expression level, but we phrase this in information theoretic terms to make it a little bit more tractable for our calculations. And then also we introduced this notion we call ICI in this thing, which is what I introduced before, that SNPs are not the same. Getting a very rare SNP is much more valuable from an identifiability perspective than a common SNP. And so we weight SNPs by their rarity measure. So we can, of course, plot all the EQTLs, and we can look at how predictable they are, and then their average, essentially their rarity or their leakage, and you get a graph that looks like this. And we can ask, how much information do they have relative to a kind of shuffle? And you can see there's a few that are kind of fairly rare, but they're actually fairly predictable. And these really are the ones that leak the most information. And then we can say, as we pick the best ones, and we sequentially pick the best ones, how much leakage do we get in bits? And obviously, if we get enough leakage in bits, we can potentially identify someone. But notice, of course, we could just mask these few SNPs, and we would, of course, get rid of all that identifiability. And we can test this in a linking attack, very similar to the one I just showed you earlier. Now, this attack's a little different. So what happens here is we have our public gene expression data set, or TF binding set, whatever, functional genomics data set, sensitive phenotype, HIV status. Now what we're going to do is we're going to create noisy genotypes, not by directly genotyping the reads from this data set, but by inverting the gene expression level with EQTLs. And then once we do that, we can match that to our query database that we've essentially stolen, just like we looked before, to link the sensitive phenotype to the identifiable individual. And we can, ask how well we can do in that context, and you can do fairly well. Again, there's additional things you can bring into the matching of person's gender, population, and so forth. But overall, you can do the matching fairly well. Okay, so that's EQTL leakage. Sorry, I have a little bit of a cough here. And then you can also do a similar thing for signal profiles. And let me show you that. And this is a little bit more tricky. So now remember, I introduced the notion of signal profiles to you. So they look like this. Now imagine if you have a big deletion in the genome. Well, it's obvious you're going to see that in the signal profile. If you have a big deletion, you don't no signal there. So obviously that kind of deletion, that 0 in the signal profile potentially leaks variant information. And likewise, often you could have a signal, you could have a tiny deletion from, say, an indel, which would also show up as just a depression around one base. And here's actually a picture of what that looks like. Here's an indel, and you can actually see it in the signal profile. And here's, of course, a big deletion. And you can see that, how it cuts out in the signal profile. So you can see some genotypic information in the signal profiles empirically. So of course, what we've went to do is to try to quantify this. We've created this formalism called private sig to do this. And it's similar to what we did before. We have a notion of leakage in bits that's related to the rarity of the genotype, as rare deletion, rare insertion, and so forth. And then we have the predictability, it's somewhat slightly similar metric to the EQTL, but a slightly different metric for predictability. We can graph the same things. And of course, there's some things that are fairly predictable that are that leak a lot of information. And then we can also think about trying to anonymize the data set by removing these most predictable bits. And then we can, of course, do a similar type of linking attack. Now, this is a little bit different, and I'm going to walk you through it again. So here, what happens is we have a number of signal profiles, and now what we're going to, and they're associated with sensitive phenotype, HIV status, and so forth. And now what we're going to do is we're going to take these signal profiles and genotype them against a known panel of structural variants. So for instance, we look at the structural variant and we say, oh, did they have it or not, because the signal is very low or very high in that position. And then we get again, a very noisy, grotty genotype of the structural variants based on the signal profile. And then of course we genotype an individual structural variants directly from the coffee cup and so forth. And then we compare them together, and then we're able to potentially link an individual to someone in this database and then get their sensitive phenotype out from that. Now, one little rub here is how we get the structural variants. Now, there's sort of two ways of doing that. One way of doing it is if we have a panel, a known panel of structural variants, which we can supply, from say 1,000 genomes, or we could imagine actually directly trying to call the structural variants from the signal profiles. This is very analogous to genotyping directly from the reads. And so we can try to call the structural variants directly from the signal profiles. And so we have two different ways of doing this, this attack. And what's kind of interesting is we find, obviously, the more variants we use in the attack, the more accurate we are in linking. But actually, that's not true in this second type of attack, where we're actually discovering the variants as well as using them for genotyping. Turns out there, as we increase after a certain amount, we actually, it hurts our linking. What's basically going on is we're just discovering noise, and we're just finding stuff that's garbage. And so you can see the noisiness in this fall off. Okay, so I think I'm going to summarize now what I've talked about. So I introduced this dilemma of genomic privacy, the fact that we want to share genomic information for medical research to get better statistical power, but potentially it's very sensitive information about an individual that they pass on to their kids and so forth. That's the general dilemma. Then I talked a little bit about the kind of tricky bits of functional genomics data, this kind of two-sided nature that sometimes we want to do studies on it that don't really have to do with the variants, and we wish we could just remove them. And also the fact that, you know, the functional genomics data is really something that we're really going to get a profusion of, because we can do essentially thousands of experiments on an individual, but only do one actual genotyping. And then I talked about the different types of leakage, you know, from the reads, from the EQTLs and the signal profiles, the major source being from the reads. And I talked about how we can quantify this with the linking attack formulation. And obviously even just a very small amount of information from the reads is significant to link an individual to a sort of gene expression or a functional genomics database. But then I showed, well, how we can do something about this, how we can sanitize the raw files in a very light way by just removing a few variants. And we can show that that they're still fairly useful in terms of this delta epsilon privacy and utility formulation. And then we can kind of have a quick estimator here, fancy, for that amount of leakage. And then I showed more subtle leakages, which leak a smaller number of variants, but are still interesting in terms of the EQTL leakage and the signal profile leakage. So now I'm going to just acknowledge the people that have helped me very much with this. So a lot of this research was led by Gamze Gurzoy, who's currently a postdoc in my lab. She led a lot of the work to do with the PBAM and the delta epsilon sort of privacy formulation for the sanitization of RNA-seq reads. A lot of the original privacy work in my lab was really started by Arif Harmanchi, who's now a faculty member in Texas, and Dov Greenbaum, who's in Israel. And we were also helped a lot just in terms of putting our software together and so forth by the ENCODE DCC, Mike Cherry, Andrew Ranker, and also Prashant, who's going to talk to you also today. And we've got a bunch of lists of a variety of our tools and websites and stuff. And we also have lots of sort of postdoc opportunities in privacy and genomics and so forth. So thank you very much for your attention. Done.
Thank you very much, Mark, for excellent talk. And you also control your time very well. So we have time for a few questions. If you are in the audience, please raise your hand first. Okay, I saw one hand raised. Let's see, who is this? I couldn't find who actually raised the hand.
I think Carl raised the hand.
Carl, okay.
All right, Carl should.
Okay, good. Do you have my audio now?
Oh, okay. Thank you. Just
Yeah.
Okay. Yeah, Carl, you can talk.
Very good. Thank you for the talk. Very interesting. I was wondering, what is the current state of the art with respect to left DNA? So if I find a coffee cup, you know, and I'm not a scientist and I'm not connected to scientists, you know, I see there are people on the web offering to like check a toothbrush for you. And I'm sure this is going to be a big progression as well. So what do you think is the state of the art and the the future?
Well, that's a very good question, because I do think that there's this whole citizen science or biohacker movement, which you guys probably all know about, that people are really, in a good way, experimenting with biotechnology, and it's getting cheaper and more affordable. And you know, I think originally this type of discussion was to some degree theoretical, and you know, people weren't going to have, you know, $1 million super sequencer in their garage. But nowadays, this is very much in the province of possible. So you can, as I was showing you, the amount of sequencing you need from a coffee cup with a standard sequencing is $20, very small amounts of sequencing. Another thing that is useful is some of the, I'm not an expert on this, but some of the technologies now are really quite impressive. Like the Oxford Nanopore has this tiny sequencing machine that you can kind of carry around and do environmental sequencing yourself. It's almost like a tricorder, you know, from Star Trek. And, you know, and I think that's a very powerful thing. And you could, you know, in some pseudoscience fiction thing, you can kind of think of you know, having one of these little tricorders and then, you know, having someone's coffee cup and getting a little bit off there, right? Maybe not in real time, but close to it. And I do think this is very possible. And I think that it's very easy to do this. And one of the points of our study was to try to deal with what we call environmental DNA as opposed to perfect DNA. Because I mean, perfect DNA, in like the 1,000 genomes database and so forth, it's much, much higher quality, the variance, than what you're going to get from a coffee cup. And so we really wanted to understand, would you get any information from a coffee cup? And one thing that's interesting, I'll just point out that I didn't show, was I think we tried also doing gene chips off a coffee cup. And those actually, ironically, don't do so well. With those, it might be the thing you might think about the most. But some of the technologies, I think, have a lot of promise for this. I don't know if I answered that well.
Thank you.
Okay, so any other questions? I think, yeah, Christopher has a question because you are the panelist. So I think you can just unmute yourself and speak. Hello?
He does not have a mic.
Yeah. So the question is the ultimate goal of developing these techniques to make these data types available in an unrestricted access manner rather than controlled?
Correct. That's a good question. I should have made that clear. So the goal of this is, I should have made that clear. So the goal of the data sanitization is that, and actually let me go back to that slide because I think that is a very important point and I probably didn't make that clear enough. So the goal of this is the following. The idea is that right now, GTEx, TCGA, PCOG, whatever, these files are locked up. They're red. They have the lock. And so the goal is that, if you believe all the stuff that we've shown you, and if you go through all this stuff, we would make the PBAM file. We remove some variants from a very small number of variants, and then we would share this openly. This would just go up on the web. It would be shared openly. And people could do calculations on it freely, like they do with ENCODE data. Now, if they really needed every last bit of data, there would be a tiny, not a terabyte scale file, a tiny little maybe megabyte scale file that they could apply for. There's the diff file. And our argument is that this would help functional genomics very much because people would mostly do processing on these PBAM files. And it's very much like, I don't know if people remember in the early days of GPS, the US military didn't want to share all the exact information on your coordinates. So they actually noised the signal a little bit, and you had to get a little more if you wanted the key to get the exact position. But people actually were very happy with using the noise signal to some degree that wasn't as precise. And so this is the idea here, that you would use this freely available PBAM file.
Okay, thank you very much, Mark, again. And I think we'll move on to the next section on the program. And okay, so let's, I think I would turn to JP for chair of the next section. Thank you.
Thanks, Haixu. Thanks a lot, Mark, for the great keynote. And thank you all for being here. I'm JP, so I'm one of the two co-founders with XiaoFeng of this event. We started, as you know, 7 years ago. So I'm very grateful to XiaoFeng of having set up the program. And we'll start with the first session. It's also great to see there are more than 90 plus participants, right? Which is one of the benefits, of course, of being online. It facilitates actually participation. So the first, we're going to have three presentations in this session. The first will be delivered by Colin Mitchell. So you want to share your screen, Colin. And I would really encourage the speakers to stick to the allocated time. So it's a total of 15 minutes, including questions. And being here in Switzerland, I'm going to be merciless in terms of time because we allow ourselves to drift. So, all right. So Colin is with University of Cambridge, Senior Policy Analyst. So Colin, the floor is yours.
Thank you, JP. Can you hear me?
Yeah.
Thank you. I'm afraid my internet connection just became unstable, so I apologize if I cut out. But I'm very pleased to be giving this presentation today on the interplay between the European Union's General Data Protection Regulation and the processing of genetic and genomic data. First, just a quick bit about who I am and where I've come from at the PHG Foundation. We're a health policy think tank and part of the University of Cambridge in the UK. We have a particular focus on genomics, data and digital health. So the topic of when and how the GDPR safeguards genomic data is potentially enormous. So in this short time, I'm going to focus on three aspects. These are the challenging questions of when genomic data are personal data, which relates to some of the risks of identifiability we've just heard about. A second challenge of how data subject rights contained in the GDPR to safeguard data apply in this context. And then I'll close with some thoughts on how we might obtain further clarity and find some ways ahead through this regulatory maze. Before that, I'm just going to give a very quick recap because I'm sure many of you are familiar with the EU General Data Protection Regulation, or GDPR. It's been enforced now for over 2 years, and it was a fundamental change to European regulation which aimed to strengthen and harmonize data protection law across Europe. Perhaps the key feature I'd like to highlight for today's purposes is that it's a sector agnostic regulation, which means that the same rules apply to genomic or health data processing as they do to social media data, for example. So that's one of the contributing factors which led to some early concerns from the health research and genomics community about how the regulation could impact the sector. So in the UK, we're funding from our Data Protection Authority, the Information Commissioner's Office. We carried out a year-long piece of research into the impact of the GDPR on genomic healthcare and research. I won't go through our methods and research questions now, but we've published a comprehensive report which considers impacts, mitigations, and ways ahead for the genomics community, and that's freely available on our website. So in this short time, I'm going to just discuss a couple of those major challenges and some of the approaches that we think could be taken to mitigate and find a way ahead. The first challenge relates to that fundamental determination of when the GDPR governs the data used in genomics. When is it identifiable personal data? Although the regulation has brought little change to the actual definition of personal data in the law, our research identified that there are considerable uncertainties still about the scope of personal data in the context and these sorts of related sub-questions. A really significant part of this is due to the fact that determining when data are sufficiently identifiable to constitute personal data is ultimately a judgment call. The recital here in the regulation sets out the broad contextual assessment that has to be made to determine whether there is a reasonable likelihood of identification. So this creates significant room for a difference of opinions. Perhaps the hardest aspect for today's purposes in relation to genomic data is the need to keep abreast of technological developments, as mentioned at the bottom of this paragraph. This is an ongoing assessment, and it could include, for example, the sorts of technical and scientific developments we've just heard about, you know, the potential for different forms of data to lead to identification, but it also includes broader availability of data, for example, the increasing availability of ancestry information, or even social media and other public sources of data that could be used to cross-reference and identify an individual. Of course, there are well-developed fields addressing this challenge. So as we're hearing a lot today, I'm sure, from a technical perspective, there are many people working on methods such as, you know, limiting the sharing of personal data by bringing analysis to the data, using techniques such as homomorphic encryption to facilitate privacy-preserving analysis of genomic data. From our perspective, there are also equally important legal and organizational measures that can be taken and that are part of the consideration of personal data in the GDPR. These include all sorts of data access controls, policies, and regulation to reduce risks of identification. But in themselves, these don't address the central challenge identified in our research, which is the uncertainty and disagreement about the scope of personal data in the context. So to address this, we think we require greater consensus on levels of risk and sufficient measures that can be applied to meet the GDPR standards in relation to genomic data and specific forms of
Okay, so we have lost our speaker. Actually, Colin informed me that he has an unstable connection, and so that he might disappear. And this is exactly what happened. So we'll wait for a few seconds. If he does not reappear, I would encourage the second speaker to warm up. Voilà, so Colin, welcome back.
I'm sorry, I'm so sorry. It's just become unstable.
It just disappeared for a few seconds, so I don't think we missed much, actually. Yeah.
Okay, well, the race is on. I'll try and finish this before the next blackout. I've turned already to the second of our challenges I was aiming to cover, and that's how the GDPR safeguards data subject rights in the genomics context. So, as many will be aware, I'm sure, there are a range of data subject rights contained between Articles 13 and 22 of the regulation, and these are designed to give data subjects some control over the processing of their data, but also to facilitate transparency and their ability to determine how data are being processed. However, there are challenges for both data subjects and data controllers understanding how they should operate. An initial one is simply the complexity and variation which influences which rights apply and how they apply to different forms of processing. That's because different legal bases for processing, for example, consent or performance of a task in the public interest, influence when rights apply and how they apply. And that's overlaid with the influence of the Article 9 conditions, which must be met to process genetic or health data, which equally allow for further tailoring or limiting of rights. So it's highly complex. A second and important question is what the specific rights require in relation to genomic data, and that can be a real challenge. So one example is, what does the principle of data accuracy and the corollary right to rectify inaccurate data require in the genomic context? Could it require some updating of older genomic records? This is a matter for open debate and interpretation. And then equally important in this domain is the question of how you manage the rights of several data subjects, for example, family members, in relation to the same genomic data. For example, how does the right of access to your data apply when that data could relate to several family members? How should this be operationalized? Does it require that you seek the views of multiple family members? How do you do that in compliance with a right not to know? And ultimately, all those involved in a collaboration who are determining the means and purposes of processing need to work out how to divide their responsibilities and explain to the individual data subjects who's responsible for which rights and which obligations. But we don't think all is lost. There is it is clear that what is needed most is clarification and guidance about how the GDPR applies to safeguard data in the genomics context. And that needs the focus of the data professional authorities in Europe. But we also think there's scope for the broader genomics and research communities to develop further clarity themselves through a twin track approach, working together with data protection experts, policymakers and regulators on specific topics to achieve both informal consensus and best practice, but most promisingly as well, perhaps to develop more formal codes of conduct or certification schemes which are supported by the GDPR itself and may be approved and relied on by data controllers under the GDPR in relation to their processing activities. And these have the potential, for example, to be used to facilitate transfers outside the EU to other international destinations, but they will not be simple to develop. So a code of conduct means that you must justify the need and demonstrate sector-wide support for your code. And if it's a pan-European code, for example, that will require approval at a European level. Essentially, we think the broader the topic of a code or certification, the harder that will be to develop for formal approval. So this may take some time and several iterations. So just quickly to conclude what I've covered really, which is that at the top level, most of the challenges we identify relate to complexity, uncertainty, and ambiguity. I picked out a couple today. Ultimately, overcoming these requires policymakers, lawyers, and everyone in the genomics community to work together in a twin-track approach, we think, both informally and to try and improve formal guidance and tailor the rules that way. Thank you very much. I'm just acknowledging here my co-authors in this project and also our support from the UK Information Commissioner's Office. I've included links to our report, and please do get in touch for any further information, but otherwise, I don't know if I've got time, but I'd have to take any questions.
Colin, congratulations, you made it in the allocated time, so yes, there is actually time for questions. So please raise your hand on the, voilà. So there's one question from Erman. There is one question also from Brad on the chat. So shall we start with Erman and then we move to Brad? I think we certainly have time for 2 questions. So Erman, I think you should.
Yeah, I can go ahead. So thanks, Colin. It was a great talk. So I'm wondering about your opinion about one thing about these regulations for the privacy preserving sharing of data. Do you think it's even possible to come up with some global countermeasures or global rules here, or do you think all these sharings should be considered case by case because the data is highly personal and the personal privacy preferences really depends on the people, right? So what?
Yeah. Thank you. I think that's a really good question. I mean, our view has tended to be, that twin track approach we're talking about, I think means that at a global level, and especially with organizations like GO4GH, there is the potential to develop like relative consensus around, reduction of risks and measures that could be put in place for certain forms of processing, certain types of transfer. So that helps to establish kind of best practice essentially. But for full kind of legally compliant behavior, that is going to have to be to a degree on a case-by-case basis, or at least, relate to swathes of processing between the EU and other countries, for example. But there will need to be a much more specific kind of focus for full certainty about legal compliance there.
Thank you.
Very good, Brad. Yeah.
So Colin, first, the report that you guys put out there is amazing. It's I really appreciate it. And the talk was really helpful. I guess I'm still looking for clarification in terms of what it seems like you've already addressed this, but I was hoping you could opine a bit further in terms of this notion of anonymous under GDPR and its relationship with genomic data. It sounds as if you're saying it's context dependent, but that seems to suggest that there are situations in which genomic data would be designated as anonymous underneath GDPR.
Yeah, so thanks, Brad. Thank you very much, because we do cover this in the report and we make our position relatively plain, I hope, there, which is, you're right, there is a debate. We're aware that some people would say genomic data are inherently identifiable, for example, and that there aren't scenarios in which you would expect to be able to reduce the risk of identification, of significant amounts of genomic information. Our view is that, yeah, we think it is feasible to limit the chance, the risk of identification sufficiently, at least in a theoretical sense. How practical that is, I mean, we're not quite in a position to say, but from a technical legal point of view, our view is very much that, yeah, it's possible.
Do you think that there will be this will require test cases and possibly, judicial review before anybody's comfortable with it?
Well, I mean, we'd hope it wouldn't work like that. And in a way, the kind of, how would I describe it? The legal order, which revolves heavily around the regulatory authorities and at the European level, the European Data Protection Board, are able to work much more kind of collaboratively with, you know, sectors and with specific institutions, and they can provide advice in that way as well. So I hope it wouldn't come to that we wouldn't necessarily need those sorts of legal challenge, but that we would be able to, you know, in an ongoing way, develop guidance for the field with the stamp of approval from the authorities.
Okay, very, very nice. So thanks a lot. Thanks a lot again, Colin. Actually, there is a lot that could be said around this. Also, the fact that actually GDPR, as you know, has different interpretations depending on which EU country you consider. So it's quite a complicated actually constellation. But thanks a lot, and thank you for reassuring us about the fact that in these Brexit times, there are still people in UK who care about GDPR. So that's great. Yeah, thank you very much. And then we'll move to the next presentation. So I think the next speaker is Kerem Ayoz, is that correct? From the University of Case Western University, and he's going to talk about something that's been mentioned before, which is kinship. So the floor is yours.
Thank you, JP. Hello, everyone. I am Kerem Ayoz. I'm a master's student in Bilkent University in Computer Science Department. And today I will present our joint work with Miray Ayşen, Erman, Ayday, and Ercument Cicek. And our title is The Effect of Kinship in Re-Identification Attacks Against Genomic Data Sharing Beacons. And in 2016, Global Alliance for Genomics and Health introduced the Beacon project. Here, the beacons are the database of human genomes, and they provide a secure and systematic way of sharing genomic data. And in this system, users can send queries about particular alleles and receive a binary response for their queries. For example, a user can ask that, is there a participant carrying nucleotide C at the 1000th position of chromosome 1, and system responds with either yes or no. Here, yes means that at least one of the participants in the beacon have that queried allele, and no means that none of the participants have that queried allele. And several studies show that this system is vulnerable to re-identification attacks, which I will describe soon. And several countermeasures have been proposed against these re-identification attacks. For example, constructing larger beacons, or sharing only some portion of the beacon, or not publishing any metadata, or having a uniform ancestor in the beacon, and or even randomly giving wrong answers to the users for some queries. But the problem is that these countermeasures decrease the utility of the beacons project. And in this study, we analyzed the effect of kinship relationships among beacon participants against these re-identification attacks, and how this might be an effective and harmless solution in terms of utility of this project. And let's start with defining re-identification attacks. The first one is called SP attack, and it was proposed in 2015, and it's based on likelihood ratio test. Here we have two different hypotheses: null hypothesis, in which victim is not in the beacon, and alternate hypothesis, in which victim is in the beacon. In the likelihood of null hypothesis, we have queries from 1 to n, and if attacker receives a yes response, which means that xi is 1, logarithm of 1 minus dn goes into sum. Here, dn is the probability that none of the n individuals has corresponding allele. And if this query response is no, which means that xi is 0, logarithm of dn goes into sum. And similarly, in the alternate hypothesis, if the query response is xi, we have 1 minus gamma dn minus 1 term goes into sum. Here, gamma is a sequencing error for that allele and queried allele, and dn minus 1 is the probability that none of the n individuals, except for the victim, has corresponding allele, since in the alternate hypothesis, we know that victim is in the beacon. And from these likelihoods, lambda value is calculated, and null hypothesis is rejected if this lambda value is less than some threshold t. And this threshold t can be found theoretically, since these are queried SNPs are independent and identical distributed. And more advanced algorithms have been proposed after SP attack. Optimal attack is one of them. Here, the optimal attack is based on minor allele frequency information of a population. And here, attacker starts from the allele which has the lowest MAF value and continues in the ascending MAF order. But here we lose the benefit of IID feature of our queried SNPs. Therefore, power calculation is based on empirical distribution, which is calculated from data. But in this attack, attacker needs much less queries. Even 5 or 10 queries are enough to identify the membership of the victim in the optimal attack. And even better algorithm, query inference attack, the QI attack, was proposed in 2018. And here, additional to the MF information, correlations between the SNPs are also used. And these correlations are based on linkage disequilibrium values. Again, attacker starts from the rarest SNP and goes into ascending MF order. But this time, by querying a single allele, attacker can obtain information about multiple alleles, since now attacker knows the correlations between them. They either both occur or they do not both occur in the victim. And even less queries are needed. 3 or 5 queries are enough to this time to determine the membership of the victim. And we use optimal and QI attack in our experiments to see the effect of having family members in the beacon. And in our experiments, we use 2 different types of datasets. The first one is the CU population of the HapMap project. We have in this dataset, we have 41 different mother, father, and child trios, but not any grandparents. And to also see the effect of having grandparents, we synthetically generate families from this CU population. We use an algorithm which is haploblock aware and it's based on minor allele frequency values. And if we look at our algorithm, it consists of 5 different parts. In the first part, child's haploblock is separated into 2 strands. Then the heterozygous alleles are shuffled to create the strands. And these strands are distributed to mothers and fathers' genome equally. And we have 1 strand in the mother and 1 strand in the father's genome. And to create the strand 2 for mother and father, we use MF information and sample by the minor allele frequency of the each allele. Then we have obtained the mother's and father's genome. And to synthesize the grandparents, we use the synthesized mother genome and synthesized father genome and give them as an input to our algorithm. And we conducted 4 different type of experiments. We see the effect of having no parents in the beacon, or having one of the parents in the beacon, or having both of the parents in the beacon, or having grandparents in the beacon. And let's move on to our results. Here you can see the power plots. In the power plots, on the x-axis we have number of queries, and on the y-axis we have the power. Power measures the power of the attacker in these re-identification attacks. For example, power being 0.6 means that attacker can identify 60% of the beacon participants, or power being 1 means that attacker can identify 100% of the beacon participants. And when we look at it, when we add mother or father only to the beacon, the power shifts to the right a little bit. And this means that attacker needs much more queries to identify the victim. But when we add both of the parents, both mother and father, we can see that the power decreases substantially and never reaches to 1 in some cases. And this result is expected since a minor allele in a child either came from father or mother. So adding both of them is quite effective in terms of damaging these attacks. And in synthetic dataset, we have a similar trend, but even more optimistic. Again, having mother or father shifts power to the right, and much more queries are needed. And also, again, having both of the parents decreases the power significantly. And when we looked at the grandparent experiment, when we look at it, you can see that when we had only single grandparent, mother's father or mother's mother, doesn't matter, the power shifts to right compared to having no family members. But having both of the grandparents, either from mother's side or father's side, has a similar type of effect with having only mother or having only father's case. But in this case, power decreased much more compared to the having only mother or having only father's case, since having both of the grandparents bring diverse to the beacon in this experiment. And we also studied the utility analysis and how adding family members affects the utility of the beacon. We defined utility loss as a fraction of additional responses that arise due to the addition of family members. And here in the table, we have control and case group. Control group are the people that are not in the beacon, and case group are the people that are in the beacon. And as I expected, we have higher utility loss in the control group since they are not in the beacon. And in the worst case scenario, we obtain a 7.96% utility loss, which is an acceptable result. And compared with the other methods, it is a much less utility loss compared with the other methods. And we also studied the MAF values of these additional yes responses. And we know that there's an inverse relationship between the variance disease odds ratio and its frequency. So SNPs with lower MAF values are much more important when in terms of the diseases. And we can see that in our plot, we have MAF threshold on the x-axis and fraction of additional yes response in the y-axis. And we can see that we have much less utility loss in the SNPs that are more valuable compared to the others that are less MAF values. So, which is a good feature of our proposed technique. And even though many countermeasures have been proposed against these re-identification attacks, new attacks have been emerging. Genome reconstruction attack is one of them. And it is our new preprint. You can check it from this QR code. And in this attack, it's possible to successfully reconstruct a substantial part of genome of a victim when attacker only knows that the victim is added to a beacon in some update. And here, in this attack, attacker takes two different snapshots of the beacon, one before victim is added to the beacon and one after victim is added to the beacon. Then using these snapshots, attacker reconstructs some partial genomes, which corresponds with the added donors among with the victim. And after that, attacker needs to identify the partial genome that corresponds to a victim. So attacker uses an auxiliary information such as simple phenotypic traits like hair color, eye color, or hair type, which can be easily obtained from, for example, social media accounts or any other else. Then, after obtaining the partial genome of a victim, attacker initiates a membership inference attack against a beacon, to another beacon, which is related with sensitive phenotype. And with this way, attacker can learn whether this victim has the disease or not by following this attack pipeline. And when we look at the results, for example, in the reconstruction results, we have number of newly-added donors in the x-axis and precision and recall values in the y-axis. We can see that precision and recall values stay high up to a certain point. For example, when we had 10 donors, including victim, we have around 0.7 precision and 0.8 recall values, which is, I think, really dangerous for the beacon users, and which proves that this attack is also a serious threat for beacon users. And as a result, we saw that beacons are vulnerable to re-identification attacks and newly proposed attacks like genome reconstruction attacks, and proposed countermeasures decrease the utility of these beacon projects substantially. And with our technique, the effects of re-identification attacks are mitigated, and even these attacks are damaged substantially. And also, one good side of our proposed technique is that we provide a symmetrical protection for both victim and relatives, since they share common alleles between them. And finally, we saw that utility of the beacon is not affected substantially compared with the other solutions. And thanks for listening. And if you have any questions, you can reach me from this email, and you can see the paper from this QR code and genome reconstruction paper from this QR code. And that's all.
Very good. Thanks, Kerem. An additional option to ask your question is to ask it now. We still have actually 2 minutes on the budget. So questions for Kerem on this talk. So I do have a question actually, which is, so can you comment on, because essentially, so you say, okay, we add the relatives into the database, right? But this also means that this was not necessarily the intention of the researchers, right? So we essentially bring more people proactively in order to mitigate specific attacks. So to what extent did you check that this is acceptable in practice in a given cohort?
Thank you. This is a good question.
Thank you.
And for example, in our proposed technique, we suggest that victim should be added with his mother or with his father or with both of them. And for example, when a child was born, a family might want to sequence their children's genome maybe in the future, and maybe they want to also sequence their genomes, and they want to add their genomes in some, for example, Beacon to store. We suggest that they shouldn't send their genomes to the beacons one by one. Our suggestion is that they should send their genomes together to some beacon. In practice, since our beacons are not that popular in the society yet, I don't see any strong examples, but I think in the future this might be a good countermeasure.
Okay. I see. So thanks a lot again, Kerem. And the third and last presentation will be delivered by Prashant. And actually Prashant is with the lab of our keynote speaker.
Hi, thank you so much, JP. And thank you so much to the organizers of GenoPri'.
Sure. So the floor is yours. So 15 minutes budget, including questions.
Sounds good. Thank you so much. So I'm actually going to shut off my video just to protect my internet bandwidth a little bit before I share my screen. Sure. So, okay. So, so thank you so much for the organizers. Today I'm going to be talking a little bit about a tool that we developed called Plight. And Plight was essentially generated to answer the question, what is the information leakage associated with a small, sparse, and noisy set of common genetic variants? How about even a single variant? So this theoretical motivation was linked to a real-world attack scenario where we considered a situation where, for example, you walked into a party, you met an individual, and you tried to see if you could play a detective game of gleaning a small set of genetic variants, especially SNPs, purely based off of somewhat innocuous sounding information like external physical characteristics that may be known to be linked to single loci, or through a slightly invasive conversation on food allergies or blood type, or as we've seen before, collecting trace genetic material from touched objects. So this brings up the question of what kinds of attacks can be done given a set of genetic variants. Now, what I've shown over here is a lot of the prior work, definitely not a comprehensive set of the excellent work that has been done in this field, but some of the attacks that are commonly carried out on genetic variant data include identity, relatedness, and I'll explain a situation about partial regional matches of an individual to others in a database that could also be used to attack the privacy of an individual. So for identity, of course, there's always the possibility of linking them to phenotypes that may be socially stigmatizing. For relatedness, of course, if either your information is in a database or your relative's information is in the database, there's a shared genetic risk associated with that. So given that we identify that these are some of the attacks that can be carried out, I'm going to present and talk a little bit about our approach to assessing the degree of leakage. So we were inspired by using genomic imputation methods that Mark touched upon briefly, which essentially fill in the most likely genotype at a given locus, conditional on a reference panel of phased genotypes, such as 1,000 genomes. And very often these methods use a hidden Markov model-based population genetics approach. And we're going to employ something similar, but instead of identifying the most likely SNP, what we actually do is we find a set, we explore the space of reference haplotypes and find piecewise matches of a query individual to maximize the information gain. So basically, this is the overall framework of the method as well as of the talk. So I'm not going to go into great detail about every component, but what the take-home message from this slide is that if you input sample-based genotypes, and if you have a publicly available panel of reference genotypes, or even a stolen panel of reference genotypes that are phased, you can set up a hidden Markov model, which refers back to the theoretical framework of a method, and the reference is back to a so-called Leon-Stephens model from 2003. But given this theoretical construction, what we do is we construct a set of piecewise haplotype pairs and carry out a whole range of downstream analyses that together we package in a program called Plight, which stands for privacy leakage through inference across genotypic HMM trajectories. So let's delve into each of the different topics there. So the Lee-Stevens hidden Markov model is a fairly straightforward model where you essentially have in the visible layer of a hidden Markov model a set of observed genotypes. And in the case of the cases we're considering, it's a very sparse set of genotypes. And the hidden layer consists of trying to match the best fit maternal and paternal haplotypes to that set of observations. The way it's done is in this Bayesian framework, where essentially what you do is you quantify the mutation error in one of the terms, and you quantify the recombination probability in another of the terms, which means that you get to explore all possible combinations of maternal and paternal haplotypes in a database of reference haplotypes. So using that as the basic construct, we use in our program the Viterbi algorithm. And again, this is fairly common as well to use. So far, it's not a particularly novel implementation. But the idea is to take these haplotype pairs for each genomic region corresponding to the queried genotype and construct this mosaic diploid genome where the numbers over here refer to the indices of haplotypes in the reference panel. And we've constructed this kind of a mosaic diploid genome from this. So the total dimensionality of the space is n times n, where n is the number of reference haplotypes. And you can, again, allow for each of these paths to independently recombine and switch over to another one of the reference haplotypes in your database. So what we also considered was the fact that given that our analysis basically is done on sparse genotypes, there's the necessity of also accounting for the fact that there's a lot of degeneracy. That is, the same query genotypes could lead to multiple possible haploid paths, diploid paths, sorry. And so what we've done is essentially accounted in the Viterbi algorithm for all equally likely paths. So in this example shown over here, segment 1 of the query genome, query chromosome, basically has several possible diploid pairs that can match. But then in this region, it becomes much more constrained and well identified, whereas in this region, it expands out again. So we call each one of these diploid paths a genotypic trajectory to signify that we're exploring this haplotype space. And within any given segment, you can sort of consider this as the entropy of your query genotypes because it seems to represent multiple possible reconstructed paths. So one of the specific ways in which we think that in which our method is novel is the fact that you could take this segment, for example, and pool information across that. So we consider one example over here. So consider the case where you have a polygenic risk score that you can calculate across all of the unique haplotypes in segment 1. So essentially you take all of those best fit haplotypes, you take the PRS scores, and then you put them against a population distribution. The idea is that if you find your population distribution falls to one end or the other of the clumping of haplotypes, it means that you've increased the risk of associating your query individual with that particular polygenic disorder. So this is one example. I'm not going to cover it extensively here, but I'm happy to discuss this at a later point. So in order to address the fact that things scale quadratically with the number of reference haplotypes, we had to come up with a few different methods. So first we have our exact Viterbi algorithm, which has high memory usage, but it doesn't make any approximations. We used a truncated version of that, which is similar to the Eagle imputation pipeline, where only those trajectories within a certain margin of the optimality are extended. And finally, the most scalable approach is this iterative module, where what we do is we essentially segment your reference database into smaller, more tractable segments. You run the exact algorithm on those, pull the results, and keep running it until you've come to a best fit set that is stable. And then you can reinitialize that randomization process multiple times. So through this bootstrapping method, we try to deal with the scale problem. So just quickly to cover some of the results that we've done, essentially the idea is that we've taken cases where we're directly trying to identify an individual in a database, where we can possibly add some mutations to it. We also want to cover the case where the individual is not known to be in a database, where what we do is we essentially randomly select 2 individuals from 1,000 genomes, splice the chromosomes, one half from one individual and the second half from the second, and try and run our algorithm on that. And finally, of course, we test against known relatives who are not in the full cohort of 1,000 genomes phase 3. So these are the results for an individual that's known to be in the database. And the most striking results from this is that even if you consider mutation rates of up to about 10%, you can often get away with just 10 SNPs, allowing you to identify the individual. Of course, as you increase the mutation or genotyping error rate, the mean and standard deviation of that goes up. But the takeaway message from this is that identification is remarkably tolerant to error rates. And what we essentially have here is, for the case where an individual is not known to the database, we splice those two genomes together. So for chromosomes 1 and 2, I present the results of our visualization module, where we clearly see that we can actually identify the two constituent individuals when we're just searching against a background of 400 reference haplotypes. And the correct individual doesn't occur in chromosome 2, but once you use a cross-chromosome comparison, you can actually gain greater information. So that's important to keep in mind. So when we expand that to a larger database of reference haplotypes, in this case, all of 1,000 genomes, it's 5,008 haplotypes, what we find is that we do pick out at least one of the correct individuals in chromosome 1, and what's not shown here is chromosome 2 picks out the other individual. So again, using cross-chromosome comparisons, you can actually pick it out from a background of 5,000 haplotypes, or 5,000 times 5,000 pairs of haplotypes. This is the example of the relatedness question that I brought up, which is essentially that you can identify definitely first order relatives quite easily. But if you for just 20 SNPs, and if you increase the SNPs to 30, you start to pick out even second order relatives. So there is considerable risk with a tiny amount of SNP information, and that's something to consider. So finally, just to actually make concrete the thought experiment that I brought up earlier, the question is, can we actually mine SNPs from a photograph, for example? So this is basically the precursor to Plight, where what we do is we use a forensics analysis for hair, eye, and skin color. And in this case, we use Susan Walsh's very interesting high-risk plex model for forensic imputation, where they are inferring the probability of phenotypes given genotypes. We just invert that task. And then we essentially look at, in a Bayesian framework, the probability of observing genotypes given phenotypes is proportional to that of the phenotypes given genotypes and the prior on that. So this is the results. Basically, the likelihood is just a product of multinomial logistic regression models. The prior is extracted from haplotype frequencies. And shown over here are, for all of the 41 SNPs in the model, the imputed values of the genotypes. So there are further considerations that I'm not going to cover here in the interest of time, but of course this could be converted to a gene level analysis that could be very interesting for certain types of disorders. But I'm happy to take any questions on that later. And I'd like to acknowledge Professor Mark Gerstein for all of his support, and Dr. Gamze Gursoy for all of her guidance in this topic, as well as many other lab members. And I'm happy to take any questions. Thank you.
Thanks, Prashant. So we do have time for, I guess, one question. So do we have questions from the audience? I think Heidi, you have a question, right? You may want to mention it. Yeah, go ahead. Actually, I have to allow you to talk.
Okay, I just wanted to point you to the Human Pangenome Reference Consortium. It seems like, you know, they're once the graph genome representations, you know, population scale mapping of haplotypes for the, you know, purpose of you know, useful biomedical data mining. But it seems like that opens up a lot of opportunities for you.
Absolutely. No, thank you so much. I mean, I think that what we saw our method was as a base model, basically trying to take things which of course wouldn't confine an individual in a database very sharply, but what it would allow is at least a carving out of space. But what you have over here, and I can see that you in the chat, you said, talked about face shape, for example. I think that absolutely could start to confine further and further. And I do know of people who are doing deep learning based kinds of genotypic imputation models as well. So it's a very helpful thing to know about this consortium. Thank you.
Yeah, feel free to get in touch with me if you want to ask any more questions about it.
Absolutely. Thank you so much. Yeah.
All right. Thanks a lot, Prashant. So the time allocated for this session is over. Thanks a lot for participating. And let me hand over to the session chair of the next session, who is, surprise, surprise, XiaoFeng.
Yeah, thank you, JP. And now we have the third session, which on the privacy preserving biomedical data computing. The first talk is from David Frolicher and from EPFL. Okay, David, you can start.
Thank you. So hello, I'm David. I'm a PhD student at EPFL in Switzerland. I'm working with Juan Troncoso-Pastoriza and Jean-Pierre Hubaux. I'm going to present our work about privacy-preserving multicentric medical research with multi-party homomorphic encryption, which was done in collaboration with the Lausanne University Hospital with Jean-Louis Rezao and Michel Coindet. So we can start by looking at a very high-level workflow of a medical research. So when a researcher wants to do a specific research, she will create a query and then check in the database of her hospital whether they have data about patients that would satisfy the subject of this study. And then she will request access to this data. So this has to go through a data access agreement between the institution and the patient. And then when the researcher gets access to the data, she can start the analysis and get results. But we can see multiple issues in this workflow. So first, if the researcher only has access to the data of her own hospital, then she might not find enough patients that satisfy the subject of the study, especially if it's a study about rare disease. Then the data access agreement can be a lengthy and costly process, which requires multiple administrative steps. And finally, after all these steps or all this workflow, the researcher might end up with data that are not suitable for the study that she wants to do, because she couldn't have access to the data before this point. In order to solve the first issue, which is the lack of data, multiple solutions have been proposed in order to do multi-site medical research. This solution can be classified in these four main categories. The first two are fully centralized and meta-analysis. In these two categories, the data are sent from the hospitals to a trusted third party. In the case of fully centralized, it's raw data. In the case of meta-analysis, it's aggregated data that are sent to this trusted third party. And then all the data are made available for the researcher. In this case, the hospitals lose control on their data, and they still have to sign a data access agreement with this trusted third party. And in order to solve this issue, decentralized solutions have been proposed. In this case, it's the algorithm which is sent to the data. So the data remain in the hospitals, and then the hospitals collaborate in order to perform a specific algorithm. But in the case of a simple decentralized solution, the information that are exchanged among the different entities can still leak some information about the patient. And that's why some secure decentralized solutions have been proposed, in which the data that are exchanged in order to perform the algorithm, so partial results, are either obfuscated or encrypted in order to protect the privacy of the patient. We can see in this graph classification of these solutions in terms of accuracy and privacy. So of course, the fully centralized, where the researcher has access to the complete data, will give very accurate results to the study. While on the other hand, for the secure decentralized solution, there is a trade-off between accuracy, privacy, and performance. As for example, if the data that are exchanged among the hospitals are obfuscated, for example, using differential privacy, then the more obfuscated the data, the more privacy is ensured for the patient, but this can alter the results of the final study. The solution we want to propose falls in this last category, except that we are relying on encryption. And we want our solution to enable the researcher to compute directly on large amount of data, which are held by multiple institutes. And we want to simplify the process as the researcher can directly compute on the data that are held by different institutes without having access to the data directly, which would also protect the privacy of the patient. In order to do that, we rely on multi-party homomorphic encryption, which combines homomorphic encryption and secure multi-party computation. So we rely on homomorphic encryption in order to compute directly on encrypted data. In this case, a ciphertext contains a vector of values, which are encrypted, and we rely on secure multi-party computation in order to design interactive protocols, which will enable the different institutes to collaborate in order to perform a specific algorithm. And if we look at the workflow of multi-party homomorphic encryption, it's pretty simple. All the institutes will each create a public key and a secret key. And then the public keys are combined in order to create a collective key. This public collective key is then used by all the institutes in order to encrypt their local results. And then all the operations are performed directly on the encrypted data. And since these data are collectively encrypted, they cannot be decrypted unless all the institutes participate in the decryption. We consider a threat model with a passive adversary, as the hospitals do not have to trust each other, but they will follow the protocol. And here is the high level or the generic workflow of our solution. So first, the researcher will do a query that is sent to all the institutes. And then the institutes, they will locally compute on their clear text data. And they will encrypt their local result under the collective key. And then these local results are combined in a collective aggregation step, still under collective encryption. If it's an iterative process, then the encrypted data are sent back to the hospitals, which will combine their local clear text data with the collectively encrypted data and perform the whole iteration again. And at the end, the final result, which is the result combined over all the institute, which is still combined under collective encryption, is switched from the public collective key to the key of the querier, which is then the only entity that is able to decrypt this final result. We can also note that only the researcher is able to decrypt this result, and that she can only decrypt the final result and not the intermediate values, as all the intermediate values are encrypted under the public collective key. Then we wanted to show that our generic solution applies for specific use cases. And so we replicated two existing peer-reviewed studies, a survival curve in which which is used to see how patients survive after a specific treatment, and a genome-wide association study, which is used to check association between specific variants and patient conditions, and which is done by training regression models for each variant and seeing if the variant has an impact on the condition of the patient. For this second example, we use a data set of almost 2,000 patients with 4 million features, so 4 million variants. In order to do the experiments, we rely on standard servers and set the parameters in order to ensure 128-bit security level. These are the specific workflow for each specific use case, so adapted from the generic workflow from before. And we can see that for the survival curve, the workflow is pretty simple. So each entity will locally compute the survival curve on its data. It will encrypt this result with the collective key. Then the results of each institute are combined under collective homomorphic encryption. And then the final combined result is switched from the collective key to the key of the querier, which is able to decrypt and generate the survival curve as if it was compute over the data of all the medical institutes. For the GWAS, the workflow is pretty similar. except that in this case, it's an iterative workflow. So the models are trained locally by each institute, and then they are combined under homomorphic encryption. The result of this combination is sent back to the institute, which will update this model using their local clear text data. And after a certain number of iterations, the final result is switched from the collective key to the key of the querier, which is able to decrypt this result. In terms of accuracy, we can see that for the survival curve, we obtain exactly the same result as if the survival curve was computed in a centralized and non-secure way. For the genome-wide association study, we obtain similar results as in the non-secure centralized approach with a small approximation, as we have to adapt the training algorithm in order to be performed under homomorphic encryption. And in terms of performance, we can see that the simple workflow of a survival curve can be done pretty fast. So in less than 2.5 seconds, if the data are split over 96 nodes. In here, most of the time is due to the communication that is required among the different institutes. And we can also note that since each hospital can compute locally on its clear text data, it means that the execution time is almost independent of the size of each hospital data set, as computing on clear text data is very lightweight. And for the GWAS, we can see that a GWAS with 4 million variants is performed in less than 1.5 hours, because we are able to parallelize the training of the models, mainly by relying on the properties of the homomorphic encryption scheme, which, as I said before, enables to encrypt vectors of values in one ciphertext, and each operation that is performed on a ciphertext is performed in parallel on all the values that are encrypted in this ciphertext. So to conclude, we propose a generic and modular solution that streamlines the process of cohort exploration and analysis by and while ensuring the data confidentiality and the privacy of the patients, and which can provide exact results, which might not be the case for solutions that rely on obfuscation of the intermediate results, as in our case, because of the use of homomorphic encryption, we are able to hide the intermediate values until the last step, in which the querier is able to decrypt the final exact results. And thank you.
Okay, thank you. And we have a few minutes for the question. So anyone raise your hand? Okay, I see Marat. Okay, allow you. Okay, unmute you. Feel free to talk, Marat.
Thanks for the talk. I have a quick question. Did you compare with other SMC alternatives? In other words, like sometimes homomorphic encryption may be slow for certain settings. So it looks like there are different alternatives that you can apply, like secret sharing based SMC to. So do you have such comparison with respect to performance and or communication, etc.? Thanks.
Yes, so we don't have comparisons yet, but what we know is that with this method is that we enable each institute to locally compute on its clear text data, which are still stored in the same institute. And so we push the computation to the institute, which can compute on their clear text data, and then they share partial results. And so the fact that they can keep their data locally also enables us to reduce the communication overhead. As in a secret sharing solution, the data would have to be split among multiple entities, which is not the case in this solution. And yes, and also our solution can work with a high number of parties, while most of SMC solution limits the number of parties that can perform the computation. Sorry.
Other questions?
So just one quick question. So I just wanted to point out one of the reasons why some of the centralized approaches are adopted is simply because of the amount of data cleaning that is necessary in order to make sure that everything is comparable. And so I know some of the previous work from your group has looked at doing quality control measures in addition to just straight up computation. Can the quality control measures be brought into this framework as well?
Yes, this quality control algorithms could be done with this solution. But of course, the problem of the heterogeneity of the data is still there, because in order for all the institutes to be able to collaborate in a specific computation, they have to understand the query. So the query they must be able to run the query on their local data. So there is a need for some homogeneity among the data.
Oh, okay. We are running out of time. So Erman, and I see you raise your hand. Could you, just understand, drop a line in the
I
chat
can ask
box?
it offline, no problem.
Okay, yeah, wonderful. Thank you. Okay, thank you, David. So the next talk will be given by Devin Enrique from Doc AI. He's going to talk about federated learning. Actually, the first talk looks very much like a federated learning, but the second one will talk about how to do federated learning and to do privacy preserving genomic data analysis. Okay.
You may be muted. I don't hear your audio.
Hi, Devin. I think you are muted.
Oh, my bad.
Yeah.
I'm Devin. I'm here with Doc AI. I'm here to talk about federated learning on genomic data. And so for context, there's utility in data. And what I mean by that is there's useful information that can be extracted from a given data set for a given context. So you might think of patients' medical records that could be used by medical professionals to give insights on how to treat them in the future, or how to treat even new patients that haven't been seen before. And so the higher the data sensitivity, the higher the privacy costs, and the less likely people are to allow their data to be used in medical research. And so something with high data sensitivity is genomic data. You might think of the utility of giving a patient, let's say, what conditions they're predisposed to, but if that same information gets into the wrong hands, it can lead to discrimination against that patient. And so we think of privacy as a first-class citizen, and we should do everything we can to protect it. And so one way we can protect data is change the way we store this sensitive information. So if we move away from centralized storage and move towards edge storage, and in this case, I mean the mobile phone, then it protects data in two ways. The first is it eliminates a single point of attack for a hacker. So if the centralized location gets breached, there's 1 million users' data in that phone, or in that centralized location, then 1 million users' data gets leaked. Now, if you move it towards the edge, that same hacker has to hack 1 million users' phones to get the same amount of information. And so the amount of effort and time to extract the same amount of information increases substantially. The second reason is, as a user, if I give my data up to a centralized location, I no longer have control of that data, and I don't get to say how it gets used, regardless of how that entity said they would use it. So if I keep it on the edge and never leaves my phone, then I'm in full control, and I get to say how it gets used. And so with this in mind, Doc AI created GeneWall, and GeneWall is the first genomic browsing app. And so what you can do is you can upload your 23andMe data onto the phone. It stays onto the phone and never leaves. And then with that, you can gain insights on your own genomic data. And so the second screenshot is showing your predisposition towards type 1 diabetes based off your SNPs. And so while that's the current state of GeneWall, we were envisioning how can we extract more information out of this data without it leaving the phone. And so we've been playing with the idea of federated learning. And so this third screenshot is a model that's actually on GeneWall. It's called Phenomenal Face. So I took a selfie and it predicted my BMI. So it predicted I'm 29, male, 5'11", 173. And so how we could add to GeneWall is use federated learning on this model. And so I would go in and update my age because I'm actually 22. My gender and height are correct, but I'm actually about 183. So I could correct that information, train a local model update, and then upload that model to a server to get aggregated with many more local model updates, which are other users who did the exact same thing as me. And the hope is after aggregation, you start converging on a model that has a higher accuracy. And so to generalize this concept, we created a federated learning mobile, or a federated mobile platform. And so agnostic to the data, agnostic to the app, you could collect data by means of the app. Users could opt into training tasks, perform local model updates, and then they could push their updates to a server and get aggregated together. And throughout this whole process, a model owner could review the aggregations and see how the model is improving over time. Now, while the data stays on the phone, it doesn't guarantee privacy. It's not enough to guarantee privacy. And so we've been adding and exploring ways to configure privacy on top of federated learning to preserve privacy, ensure that the user's privacy is preserved. And so one aspect we're researching is how does the model structure change or affect the amount of information that can get leaked through a model? Another one, similar to the last talk, is secure aggregation. And what that does is basically you mask the model updates and it only gets revealed once all the model updates are combined together. And that prevents people on a server or the server itself from inspecting local model updates and preventing like a gradient attack, which could reveal specifics about the local data that it was trained on. And then another huge layer of privacy is differential privacy. And you can think of that as adding noise to the model to mask or prevent the model from learning the specifics about a user's data, but not enough noise to prevent the model from learning in general, what do all users or all models have in common. And so it's a way of ensuring generality of the model without overfitting to users' data. And so we wanted to create a model evaluation and admin UI that anyone could create tasks, upload those tasks, and have actual users participate and use their own data on. And throughout this whole process, we wanted to be able to track the progress of those tasks. And so you can see at the bottom, it says current epsilon of 0.33. So this model had differential privacy added to it as a layer. And after an actual aggregation round on Phenomenal Face, this was the epsilon. And you can think of epsilon as a mathematical way to kind of show how much information potentially could get leaked. And so in GeneWall, we wanted to come up with an actual genomic experiment where we could add it to GeneWall, like upload genomic data to it and run a federated learning task on it. So the premise that we came up with is, what if you got a biopsy of a tumor, you got back the microarray of gene expressions, and that would tell you which genes are on or off, basically, and then upload that into GeneWall. And since you got this biopsy done, you would know whether it's cancerous or a normal tissue. You could import that into your GeneWall and opt into a federated learning task. And so to simulate this, we used the Ayday 2019 data set, had roughly 18,000 genes. It used 470 breast tissue samples, 48 were normal, 422 were cancerous. And then we also did since it was a fairly small data set and we didn't have a validation data set, we did a 5K fold cross-validation on it. And so every line there is one run of the data set with federated learning. And then 1/5 of that data set was held out for validation, 4/5 were used for the training. And so within any given round, we wanted to sample, because in a federated learning scenario, you might not use every client in a given round. So we sampled 30% of those 4/5 of the data into the each round. And then we also took the assumption that one user of GeneWall would upload one biopsy. So each model was trained on one, each local model update was trained on one sample. And so you can see that in the top right corner that it actually learned in a federated learning standpoint that we could predict whether or not a tumor is cancerous or not. And you can see that the model actually converged to a high accuracy. And so if you guessed 1 on this data set and just said everything is cancerous, you get a baseline of 90%. And on a centralized training, you get about a 99%. And so you can see that by removing the centralized storage and moving it on the edge, you're not really missing out on that much accuracy. And we wanted to take it a step further and test, okay, now what happens when we add differential privacy? And you see 4 out of the 5 runs, the noise wasn't enough to prevent learning. One run, it knocked it out of the learning phase, but 4/5, it had comparable results to just regular federated learning. And so what were the takeaways here? So 1, GeneWall is the first genomic browsing app. However, we wanted to utilize that information that's stored on the phone and expand on it to extract more. And so we explored the idea of utilizing federated learning to extract more information. However, we didn't want it for a specific task. We wanted a broad platform that's agnostic to the data, agnostic to the app, that anyone could employ and accelerate medical research. And so by doing this, this project, we gained privacy-preserving insights, and we now have a better understanding of how to preserve people's privacy in a federated learning scenario. And our goals is to increase data availability by reducing privacy costs associated with extracting information. And our hopes is that by deploying these on apps, we accelerate medical research because more people are involved and aren't afraid of their data getting leaked. And so we're also looking for partners who want to join us in these efforts to accelerate medical research with an emphasis on privacy. And if you want to know more specifically about GeneWall, you can, I have a colleague who's giving a talk at ASHG. And if you have more questions of how you can get involved with us or more about this talk in general, there's info@doc8i. And I'm happy to take questions now.
Okay, we have 5 minutes for question.
Well, I can go ahead and ask one. Did you, for epsilon, how did you choose your epsilon for the task? Because for differential privacy, it's very important thing. And I'm very curious that because different parties are doing this in a privacy preserving setting, and if you don't do with the right epsilon, how would you know you get a good model? In other words, how would you test for epsilon? How do you set it in your application domain?
So that's very task dependent. So, and based on the privacy needs, the best way to test is just to perform attacks on it. But we do multiple runs to see, you can simulate differential privacy with different epsilons and see how does the accuracy change, and it's kind of a trade-off between how much epsilon you add or how much noise you add and what's the accuracy. And depending on the task, like this task, we're allowed to keep a lot of noise, and that's because the decision boundary was so great. It wasn't so small that a little bit of noise would knock it out. So it's really task dependent.
Other question?
Yep.
I have one if nobody has. So actually it's a follow-up question to Murat's question. So you said privacy needs, right? So how do you define privacy here? So what type of attacks are you considering? For example, the differential privacy provides you some privacy, but it depends on the types of attacks that you do and the value of the epsilon, as Murat said. So are you trying, for example, model inversion attacks or any type of inference attacks here?
So membership inference is one that we're looking at. You wouldn't want to reveal who was actually in the training set, especially with genomics data. The other one is model inversion. Those are the 2 biggest attacks. There's lots of other attacks we researched. Also grading attack, but we prevent that with secure aggregation. So the 2 main ones we're looking at is model inversion and membership inference.
Okay, so that selected value of epsilon, could you experimentally show that it mitigates these attacks that you consider?
Right, and you can do different, like train the same model on the data set with different values of epsilon, and then see perform the attack and see how successful it is based off different epsilons. And one thing that we did come up with, or not come up with, but we found is that if you change the model structure, it also reduces the likelihood of those attacks. So if you add skip connections and a different type of structure, so that can also reduce the amount of noise that you need to add to prevent these attacks. So it's the idea is that you can add different layers and they synergistically work together to protect users' data.
Okay, other questions? Okay, actually, I have a question. And during federated learning, right? So I guess you guys using those Google style approach, and you guys exchange gradient, right? And multi-party exchange gradient, and also add the differential privacy noise to the gradient, right?
Right.
And I think in doing so, have you measured the performance impact? And have you really do these things across multiple phones? so I could, just and have you guys, did you do the experiment just in simulation or just do it on the real system?
Right. So the that gene wall task I showed in the admin UI, that was an actual run on a phone. Mm-hmm. For the simulation of the breast tumors, we did that on just on our laptops on a server. And so
okay, so go ahead. So is there any performance? So what is the performance and across the phone? I think the GWise one is looks much more data intensive than the other one, right? And have you measured the performance?
Which one's more intensive?
GWise, the GWise one.
Oh yeah, so we are collecting a data set to evaluate that. But we've also done previous experiments on with different data sets and see how the different epsilons change. And then we're also testing, like you can change how you apply the differential privacy, like per layer, global. You can change if it's a global differential privacy or local. You can add it in the optimizer, or you can add it after the model has made its gradients. And so we're testing all that and see which gives the best performance with the best privacy.
Okay, thank you. And could you kindly unshare your screen?
Yep.
Thank you. So the last talk is about another way to achieve privacy preserving data analysis. And this time, and I think the presenter going to talk about using hardware supported confidential computing using Intel's SGX. So the speaker is Ko Dokama from Indiana University. Could you share your screen?
Thank you. Can you hear me well?
Yeah, I can hear.
And the slide, it's up and going?
Yeah, I can see. Perfect. Go ahead.
Okay, so one sec. Okay. As XiaoFeng introduced us, we would like to present our work on using Intel SGX secure hardware assumption to do privacy preserving genotype imputation. So I am from Indiana University. I'm collaborating with my colleagues from IU, from the National Cancer Institute, and from the Broad Institute of MIT and Harvard. We are based in the USA. So we would like to offer a solution to genotype imputation. So let's start with what is genotype imputation. Now, lots of panelists have talked about what this is, so I'm going to be brief about it. So genotype imputation is the process of statistically inferring unobserved genotypes from known haplotypes in the population. And when you impute, you assume that if one particular SNP is present, then a certain number of other SNP should be present as well. So effectively, imputation replaces missing genotypes by basing results on existing information. This helps by reducing the cost of sequencing DNA, and it helps with improving downstream analysis like GWAS. So the algorithm that we are basing on is called Minimax.
Hello, Ko. We see your presenter view.
Oh,
not just the slides. Yeah.
Yeah, I think you shared the wrong screen.
I shared the right one. It's, yeah, what is the problem? I'm so sorry. So let me do this instead.
Yeah.
Okay.
Yeah, now it's fine. Go ahead.
All right, so the algorithm we are basing on is called Minimac, and it's one of the state-of-the-art algorithm based on hidden Markov model. And this is currently being used in servers like the Michigan Invitation Server. So, and other panelists also mentioned how this works in deep detail. So I'm just gonna mention like why this algorithm works so well. And that's because, first of all, the hidden Markov model is a sequential model. Second of all, the hidden Markov state here captures the ancestral DNA recombination process whose fragments are assumed to appear in the reference genomes here. And there's another thing that's being captured here, which is the emission process, right? And this is supposed to capture the mutation in sequencing error. So Minimac, based on hidden Markov model, is very natural to the imputation method that we would like to do in SGX. So the way it's being done right now is that in servers like the Michigan Invitation Server, users are uploading data in plain text to the server, and the server has the reference panel locally. So there are, and this works fine, but if you have a different TET model where the leakage can come from the plain text, then this raises a privacy concern, right? So this concern has been raised by the Ayday competition last year in track 2. Which challenges participants to use homomorphic encryption to do genotype imputation in linear model. Now we want to do something different. We want to get closer to the Minimac to get a better performance. We also want it to run faster in real time. So how do we do that? The basic idea and the simple way to do that is to put Minimac in SGX. And what SGX is offering is hardware protection. Which basically translate to memory encryption and protection against probing into the CPU. So this works fine. Well, also that user can go through the process called remote attestation to verify the code base that is being run to make sure that malicious code is not being run there. And after that, they can establish a secure tunnel to feed in input and receive outputs. So this works fine, but there are some set of challenges here. There is the memory limitation that is inherent to SGX. There is slow memory access due to memory encryption, some side channels, and other class of attacks that we are not going to be addressing in this talk or in this work because it's much harder to deploy and also cost much more. So we're going to be addressing memory access pattern and the timing attack. So there are many attacks that we have been trying. This is one of them, and we're going to demonstrate how it works here. So this attack is combining 3 different attacks in literature called floating point timing, port contention, and microscope. The first one is to extract the side channel. So how it works is that in this loop, in the emission function in the hidden Markov model, there is a user genotype here that is being compared to the reference rate. And this branching can cause timing discrepancy between branch A and branch B. When the attacker can control how large or small they are, multiplication can take a different amount of time, and that's how the attacker would start. Now, in order to be able to observe how long it takes, the attacker can spawn a monitor process to monitor the floating point unit. So, and the last one, because we want to keep repeating this process to amplify the signal, we launch a microscope attack to cause page fault at a memory address to trigger the CPU to run speculative execution over and over again over the same code. And if you have heard of speculative execution before, it's responsible for Spectre and Meltdown attack. It is a spatial optimization in Intel CPU to predict the execution before it is actually doing execution to save time in the future. So with this class of attack, we have our results here, and each section here on the y-axis we have the number of clock cycle it takes. So more mean it takes more time to compute, and less mean less time. So this corresponds to our secret bit, right? And each section take about 1 second. Each second is one exact loop that is trying to extract one secret bit. So we run this experiment on a modified Linux kernel to capture the page fault. And we use the Gaussian mixture model clustering analysis method to cluster between 0 and 1. And we get about 92% accuracy, and it takes about 1.5 seconds to extract one bit. And to remind you here that each bit is a user genotype. So it could take a day to extract all the inputs you feed to the system, even if you're using SGX, to remind you. So how do we address this problem if we want to use SGX? What we do is we refer to the existing literature that points out the leakage. Floating point operations are pretty much out of the game because they are too leaky. Also, integer division is also too leaky as well. You also don't want to use if and else because that the branching leaks information. So we want to replace that with safe operations that have been proven to be leak resistant, including integer addition, subtraction, multiplication, and other bit operations. So because of this limitation, we have to come up with a few strategies. The first strategy is that we want to use multiplexer circuit to do the switching. And this multiplexer makes sure that both branch A and branch B are being computed before they are being fed into the conditioning. So that's the first one. The second one is because we cannot use any floating point primitives, we have to use integer primitives. And the way we do that is using a fixed point representation. So if you don't know what a fixed point representation is, you basically scale up the real number to integer, and then you represent that with integer. So now you can use the safe operations that are being allowed in this subset. So, but now the problem is when you're representing numbers with fixed point, you are losing a lot of precisions because it's not the most efficient way to represent, right? We have to do a lot of transformation to be able to preserve the smallest number. Now there is a side effect to doing log transformation, of course, because multiplication turns into addition and division turns into subtraction, right? But how are we going to do addition and subtraction in log space? This is one of the challenges that we have to go through. So what we do is we have to be able to compute this function on the right here, which is exponentiating addition and logarithmic. So we call this log sum exponent, and log diff exponent is the counterpart to that. And we have to do this with the safe operations, right? So what we do is we are breaking LSE, for example, into two components. The first component is easy to do. The second component requires some piecewise polynomial approximation. And this is possible because if you look at the graph here, you can see that it converges to 0 very, very quickly. So you don't really have to approximate things that after a certain point, and we just have to break the parts that are relevant into multiple sections. So here's our implementation. We're doing this in Rust and using 410x EDP as an Intel SGX SDK. We use the previously described technique in our implementation. We also enforcing static typing to ensure secure operations. And what I mean by that is that when the safe type is touching the unsafe type, it forces the unsafe type to change into safe type. And the only way that you can go back from safe to unsafe is to be explicit about it. So now be sure that we don't miss any part in our code. So here's the result for accuracy. We are using 1000 genome as a reference panel and chromosome 20 as an input. So we only take 1 out of 3 chunks because that's the way that Minimac is doing. It's just splitting the inputs into 3 different parts and work on them separately. So we sample 100 individuals from 1000 genome, use the rest as a reference panel, and here's our result. So just to explain what this is, the y-axis here is the accuracy, is the R-squared value, and the x-axis is the minor allele frequency bins. So our result indicate that we have an identical solution to Minimac with whose difference is actually statistically insignificant. So our solution is in every way equivalent to Minimac. And here's the benchmark. So let me walk you through this graph. The first one is running Minimac in C++. So it's taking about 12 seconds to run Minimac, and it's taking 5 GB of memory. But then we, So our implementation of Minimac in Rust take only 2 seconds and 20 MB RAM. So the baseline implementation is already better. Now we throw that in SGX, it takes 8 seconds and 5 megabytes. So now SMAC, secure minimap, take 7 seconds outside SGX, and inside SGX is taking about 24 seconds, which is twice as slow as the baseline. So there's a bit of a jump when you go from non-SGX to SGX about 4 times, and this is the gap we are trying to close in the future work. So finally, we want to offer you 3 different solutions, right? We have the no privacy solution, which is faster than the Minimac code. We have the leaky solution in SGX, which can work well if you know that there's not going to be any type of sophisticated attack on your SGX. And finally, we have the leak-resistant solution in SGX. And in future work, we plan to close this gap even further to make this not an alternative but a replacement to existing solution. So finally, I would like to thank my colleague for contributing to this work, and I would like to leave it to the Q&A with the final slide. Thank you.
Thank you. And any questions? So we have 3 minutes for question.
I have a question. This is Murat. Coming back, so because of the speculative execution, you identify certain operations as having speculative execution in it, so you try to change them, right? So everything else doesn't have any speculative execution, even if loops have that, right? Sometimes. Speculative. I mean, jumps have also speculative execution. So all I'm saying is that can you just solve the speculative execution problem by focusing on few floating point operations? So I think it's deeper than that, and you have to disable all of it. So does it make sense what I'm asking? Like, I'm not sure.
Yeah.
The threat model here.
But the reason why, speculative execution is a vulnerability, it's also an optimization. So by getting rid of it, it makes your CPU go slower. So there is a trade-off with that. And the reason why you don't really want to get rid of that is because it affects everything else that you're running on your server. So it's better to address that from upstream than to take away some of these optimizations that can really hurt you overall.
But my point was that, like, if you just address these leaky floating points, I'm not so sure whether you address the entire vulnerability of SGX to these kind of microarchitecture attacks. So will that be enough? That's all I'm saying. Or
okay.
Also attacks possible because of the speculative execution anyway.
So our solution address all the existing attacks in literature. That includes so there is the memory access pattern. That's another big thing, right? And that our program is already safe against that because there's no memory access pattern because the input is sequential. So so the short answer to that is that our solution can already address existing class of software attack in literature. So if yeah, I don't know if that answers the question, but it's always it's always the attack against the solution. So the solutions have to keep improving as it goes as well. But what I can say right now is that this is secure against all the existing, or most of the existing class of attacks out there.
I'll let others ask question.
Other questions? Yeah, and I would like to further comment on Murat's and question. I think and Cole, you need to be very careful about you addressing all existing problem. And there are a lot of microarchitecture attack. For example, you have MDS attack. There are a lot of buffer if you're running on the same core, right? If people hyper-threading running on the same core, and even you are running on different core, and the microarchitecture buffer could leak out information. And I think your approach, I don't think it address this type of information leak. Just if you have a data which keeping store buffer in node buffer and then another process running on the same core, and they have a lot of way to get a data which you keep on the microarchitecture buffer, and your approach apparently does not defend against those threats.
Right, you are referring to something similar to a foreshadow attack, something like that, right?
A foreshadow is on the just out of order execution. I'm talking about attack just like and a redo and IVL and and Zombie node and those attack.
Yeah. Yeah, please.
Yeah,
Oh,
I don't
okay.
have the impression you can address all this attack.
Oh yeah. So
my only comment is that sorry to interrupt. Is that like Microsoft has this confidential computing cloud that they are using as GX, and they claim to have a setup they prevent some of these known architecture attacks. That's the reason I think it may be good to compare your solution, what you are protecting, versus these server changes or CPU changes that try to prevent against those attacks, and look at whether how the performance impacted. That's my final
I think you're referring to Microsoft Azure Server, right? And I think we are talking about the same thing. They're talking about SEX to address.
But my understanding is that at kernel level, they do some changes to prevent some of these attacks, so that hopefully some of these for-shell things are not attacked. So therefore, if you do the memory access control combined with these kernel level changes, they claim to have some protection against those attacks.
Yeah.
So I was kind of seeing is that kernel level protections versus this kind of protection, how do they compare?
Yeah, and we have to stop, and we're already running out of time. And yeah, so I think that's the end of these sessions. And the next one will be our panel. And Brad Mannon will be the panel chair. And Brad, 45 minutes, your turn.
Thanks, XiaoFeng. Okay, so let me share. All right, so it's time for something completely different. So when this workshop was started, about 7 years ago now, and one of the goals was to try and cross boundaries between the computational world and I'd say the more policy-oriented and social world. And so this panel is meant to give an illustration of how this is possible. It's going to be very different than anything else we've seen today. So the whole point of this, where this is coming from, is about 5 years ago now, we started what we refer to as a transdisciplinary center. And we did so with the assistance of the National Institutes of Health, the National Human Genome Research Institute, through what's called the CERE program, or the Centers of Excellence in Ethical, Legal, and Social Implications Research. And so this is something that on Wright-Clayton and I put together, which was what became known as the Center for Genetic Privacy and Identity in Community Settings, or GetPrecise. And if you want to know more about it, you can go to this website. But it's meant to be an environment that is taking a more holistic view on the challenges associated with genomics and these issues of privacy and personal and community identity. And what we did was we brought together experts from many different domains, people with backgrounds in anthropology, communications, computer science, economics, English, genetics, law, sociology, medicine. I can keep going on and on, but you can imagine that one of the challenges that we've always run into is trying to find common ground and a common language for getting everybody to speak with each other. So we try to create very broad problems that can draw people together to work on these topics. And so one of the recent thrusts has been how to understand people's personal perspectives on the matters that I've just alluded to, and not just try to solve problems and then ask people if they want the solutions. And so what you're going to see in this panel is from several different perspectives. One is this notion on how people bring about the genomic issue from the perspective of literature and fiction in particular, and that's what Jay Clayton will be talking about. And then Jijun Yin will take a look at how people naturally talk about issues around genomics and particularly in the direct-to-consumer setting with respect to online social media. And then Catherine will be talking about how people feel about genomic information being collected and used, and how it's collected in the context of research from the LGBTQIA+ perspective. And then Ellen's going to bring it home by talking about how this information is collected and used within the context of the clinic. So I'm gonna pause here and turn it over to Jay once I stop sharing.
Thank you, Brad. Can you hear me? Thank you. So today I'm going to describe the work our transdisciplinary team of researchers are doing. Our team is focused on uncovering how culture affects people's attitudes toward genomic privacy. This is an important endeavor. More people are exposed to issues posed by genetics in popular culture than in any other space. Participating in this project has helped me, got to get my cursor in the right area, has helped me articulate a methodology that can enable humanists to contribute to such an important project. As Brad mentioned, the key to this methodology is that we're focused on a single broad problem in which researchers from a variety of disciplines, in this case genetic privacy, focus on a single issue, genetic privacy, from diverse perspectives. In most humanities research, scholars choose their own topics and work in isolation to answer questions that they themselves think are important. By contrast, our group addresses an issue that is widely perceived by people outside the academy as a pressing social concern. Our method should be described as transdisciplinary rather than interdisciplinary because it's addressed by ad hoc teams of scholars, and each researcher brings his or her specialty to bear on a shared problem. We share data and findings, but we use our own research methods. In my case, that would be close reading, film theory, gender and race studies, and more. But the group as a whole has regular meetings to discuss progress and results. We publish focused studies in disciplinary specific venues, but we build towards synthetic conclusions and policy recommendations. To investigate how culture reflects and informs public attitudes toward genetics, our team of researchers has collected the most comprehensive database ever assembled of literature, film, and television shows about genetics. It consists of 800 plus unique items dating all the way back to 1912, and we're still updating the database every week with current movies in 2020. We've coded the data in this data set for 109 different attributes. We assembled our database by searching descriptions of media from sources such as IMDb, Wikipedia, and much more specialized film and television data banks. And we supplemented our collection with hand searches of citations from film and television criticism and from films that we knew about from our own viewing experience. 2 independent researchers tagged each record for 109 different attributes, categorizing it by genre, medium, attitude towards genetics, scientific motifs that are featured in the films and television, and 16 different perspectives on privacy and surveillance. In addition, we harvested all the scripts or subtitles for movies that we could find. This came to about 120 in all. And we're working with our next speaker, Zijun Yin, and his research assistant, Kaning Ni, to analyze these texts using topic modeling and sentiment analysis. The novelty of this process lies in our method of using our transdisciplinary approach to cross-validate the research results obtained from other approaches. This process can be visualized as a cycle in which the items collected, tagged, and analyzed statistically will provide the data for sentiment and topic analysis, which will then be interpreted at a far closer scale under the lens of humanities-based close reading. If our close readings confirm the insights of sentiment and topic analysis, then we will have generated a novel method for generalizing the insights of humanities interpretation. None of the 3 disciplinary approaches will have to stand alone. Each will have its result tested by the other disciplines. The close analysis cover representative works in each of the 7 genres that we have identified as being prominent in film and television depictions of genetic privacy. These genres are of the sort that you would see in a video store were such a thing still to exist, but they're also the ones that have the largest number of films that feature genetics in them. Science fiction, melodrama, which are generally family and personally based stories. One of them was mentioned earlier this morning, the Immortal Life of Henrietta Lacks. It would be a good example of a melodrama about genetics. Superhero films, that often made from comic book superheroes, horror, conspiracy thrillers, dystopia, realistic stories about genetic decision making. So we selected key texts from each of these 7 genres and have done very close readings of them. First of all, we started with a quantitative analysis of the bioethical concerns that emerge in the different decades of film and TV. That is, we were able to look at how film and TV took up genetics in different ways over the many decades that filmmakers have been attending to this topic. And we found some amazing things in our research. We found echoes of Nazi eugenics in all the Marvel X-Men films. We've found the shift from personal privacy concerns to more informational concerns surrounding data privacy to be really prominent in the shift between the 3 versions of the Blade Runner narrative. The urgency of privacy concerns in LGBTQIA+ communities in Orphan Black has been a very interesting topic to explore. Privacy breaches in medical dramas such as ER and House. We surveyed all 65 television series of medical dramas, over 5,000 episodes of medical dramas that we looked at, and selected all of them that dealt with privacy questions. And you can see a few more topics that we've published papers on, but I have 30 seconds left to round out this talk, so I better thank my great team of researchers and thank you all for your patience in listening to something so far out of the area of computer and bioinformatics and computer science.
Thank you, Jay. We're gonna hold questions to the end. So we are gonna march along, and so Jejun, I'm gonna ask you to grab the screen.
Okay, can you hear me?
Yep.
Cool. Okay, hello everyone. This is Zhijun Yin. Today I'm going to talk about learning direct-to-consumer genetic testing user experience via online discussion. Via online discussion. So, what does direct-to-consumer mean, right? So, it means that a genetic testing can be completed without any healthcare providers. For example, we can take some issue sample and send the sample to a genetic testing company and learn about our ancestry composition, genetic health risks for certain health issues, and DNA relatives. DNA relatives is a service that can help find out testers who share similar genomic information with us. There are millions of consumers already underwent direct-to-consumer genetic testing, and Ancestry DNA and 23andMe are the 2 companies that cover the majority of consumers in the market. So previous study already showed that while it has a potential to learn a person's identity and health risk, the results of direct-to-consumer genetic testing may be not accurate, and it can vary across different testing platforms. Additionally, the raw data of direct-to-consumer genetic testing can be requested by the consumers, and the consumers are free to upload their information to the third party, any third party that they want. For example, the GDPR, right? So I think perhaps the most famous case related to genetic tests and related to the GDPR is that the information stored in GDPR were leveraged to track down the golden state killer, which is very interesting and fantastic, right? However, the data uploaded to GDPR can also disclose the genomic information about tens of millions. You know, even if those people never underwent any direct-to-consumer genetic testing or consented to sharing their information, right? This created concerns about privacy in children. However, how consumers respond to the service in general, especially after obtaining the testing results, is incomplete. Also, the insights provided by research to date are further limited because they are based on surveys and interviews with consumers. These surveys and interviews reveal response that may be shaped by the investigators' questions as well as the hindsight and bias, rather than the respondents' unpromoted reports of their own experience. So given the prevalence of internet, online social media platforms provide opportunities to learn about what aspects of direct-to-consumer genetic tests are interesting to consumers by examining what they choose to say either on their own or in conversation with others. It should be noted that there are several studies using Twitter data to learn what people say about their testing results. However, we still believe that they are limited because Twitter is designed for, it's a social media platform for the general discussion. It's not easy for consumers to develop deeper discussion on the related topics. So in our study, we collected data published by more than 16,000 Reddit users in 23andMe and ancestry DNA to subreddits. We then applied a computational method to infer and examine the topics discussed in these two subreddits, and also the temporal trends in posting rates and themes. The themes are the aggregation of related topics and also the emotions expressed in these two online forums. So here are our main findings here. First, we found that the posting rate increased sharply after popular promotion events such as Black Friday and also the Amazon days. Our topic analysis showed that the related topics are about tracking testing progress. For example, from submitting a sample to the genetic testing company until the obtaining the results from the companies. Second, we found that the ancestry composition and kinship feelings were the two most frequently discussed themes in these two subreddits. Particularly, we also found that there is an increasing post talking about it from two different aspects. The first aspect is that some testers are trying to find out their biological relatives. On the other hand, some testers who did a relative matching, who did the DNA relative matching, found out that their current family members are not their biological ones, which led to a great shock in their life. Third, we found that discussions about health risks in these two different platforms were less common, but focused primarily on how to submit the raw data they obtained from basic surveys to third parties for health risk interpretations. I think this is very interesting because, especially considering that the health risk service is not included in the basic service for many companies, and you have to pay it in order to get these tests. So for more details, you can refer to our recent publication in PAS 1, which is done with my colleagues like Dr. Li Jun Song and Alan Clayton, and also Brad Malin. That's all my talk today. Thank you.
Perfect. All right, we are right on time. So Catherine, feel free to grab the screen.
All right. Can y'all see me? Can you see my screen?
We can.
Awesome, thank you. So I'm Catherine Hammack-Avaran. I am a lawyer by training who does a lot of qualitative research around genetics and health. And one of my favorite things to study is what people think about whenever they are making decisions or receiving things like genomic results. So I'm going to just sort of give you just a little taste of some preliminary results from two research studies that we are actively analyzing right now. So the first, we're jumping into LGBTQ perspectives on genomic research. First thing I want to get clear from the start is when I say gender, what I mean is the gender that I am, that might be how I feel, how I identify, how I present. And when I talk about sexuality or sexual orientation, that's whoever I'm attracted to. So we asked research participants to imagine this totally hypothetical precision medicine research study, which we called the National Research Project, which may look familiar to some of you as sharing a lot of characteristics of the All of Us research program. So that involves collecting biospecimens and genomic analysis, but also includes things like medical record data from like health records and lifestyle information, so maybe from surveys, things like that. So of course, the point of this is to create one central database for all kinds of researchers to use for many years to come. So we asked, we sort of set up this hypothetical as being, maybe they're investigating health, but maybe they're also investigating LGBTQ participants and trying to inform a question about, the extent to which there is a genetic contribution to one's LGBTQ identity. So in other words, this is sort of a revival of that 1990s gay gene research. So when people signed up for this National Research Project, perhaps that was in their mind that maybe that might happen, but that wasn't the purpose when they signed up, but it's happening now. So we asked them to sort of talk to us about what that would mean for you. So one of the biggest topics was the extent to which this has any value. So while some people said, yes, I've known for my whole life that it was very different, and so this would help me and help our community to teach us why we are the way that we are. Most people said this just isn't valuable. So, this person says, I don't think we need a marker to justify our identities. I don't need to find out if there's a gene or not. But we asked them, okay, let's say that researchers do this type of research, and they do find that, yes, there is some genetic contribution. We asked what that would mean to people, and a lot of people talked about validation. So this person tried not to cry and failed there, but said, I've asked myself this question my whole life, like, why am I such an outcast? And clearly this person came from an environment that also contributed to questioning why they were different. And so to know why they were different would be really helpful for them. But other people said sort of the opposite. So one person said, as somebody who's already lost everything to come out, I would be terrified that what if they did find a gene, I didn't have it? And that would make me feel like less of a gay person. Another person references the Dr. Seuss book, Sneetches on Beaches, where we have the sneetch, the star-bellied sneetch situation, which is where some people in the LGBTQ community have the gene, right? They have the star. They're the special ones, and so it might throw off this this dynamic within the community and add to already sort of discrimination and exclusion within the community. So people also talked about the risk of pathologizing this type of result. So this person, and not the only one, said I'd be nervous about people thinking that there's some sort of variation means that there's something wrong with them. Of course, they also talked about eugenics, which some of them talked about, editing out of queer people who exist now. So maybe trying to do gene therapy, right, to fix them. And people also talked about editing out of generations yet to come. So this person said, well, if it's genetic and I have it, then maybe I might pass it on to my children. And then if people don't want gay people in the world, then they're going to tell me I can't have children. Related to that, of course, is this idea of determinism. Some people said, listen, even if it's genetic, even if it is sort of predetermined in some way, I don't think that that would mean anything to me because my identity is the product of a very long process. So we also asked them, what if there was no genetic contribution? Of course, people said almost the opposites, right? So maybe that might be invalidating, but maybe it might be empowering. So this person says, it would be liberating to me because then if we can define what our community is and who's included, then we can use that to our advantage. We can be more inclusive, and it can be empowering. So in the end, we asked these people, you know, this kind of research, is this acceptable or not? And there was about a 50-50 split. So some people said, you know, maybe for the people who said it's acceptable, maybe for health reasons, for medical decision making, that might be helpful. But other people said, no, it's like playing with fire. The risks are far beyond any potential health benefits. So in the interest of time, I'm going to, I always blab a little longer than I think I will. I'm going to skip through. We also asked a totally separate group of people in focus groups in Nashville to talk about, you know, their ideas about direct-to-consumer genetic testing. And one of the things that we've specifically asked them about, if I can get all the way through, why would I think I could do this, is law enforcement access to data. So once a direct-to-consumer genetic commercial company has my data, can law enforcement access it? Would I be okay with that? And in the end, there is a pretty even spread. So some people really focused on this idea if they can help someone, that would be a positive in their mind. This person says, we all have that one family member we never trust. We know that they've done something wrong. We just don't talk about it. And in explaining, you know, this is, I would want for them to be brought to the legal system. Other people said I would not be likely to share that. So if you know your family has a less than beautiful reputation, you don't want to hurt your tribe. They might not be great, but they're still my people. And other people said things like law enforcement aren't always the good guys, as we are seeing. This person was concerned about nefarious uses or questioning this idea of the greater good for everybody. And of course, some people felt clearly in the middle and said, you know, I'm trying to balance this. I wouldn't want for a murderer to get off, but I also wouldn't want to ruin someone's life unnecessarily. And so I'm torn. So just under, I think. So this isn't sort of meant to reflect all of the things that people talk about, not meant to reflect all of the things that they think about. Just a sneak peek on the types of things that people are considering and how they perceive the meaning of their genes and their genomic data. So thanks.
Thank you. I wish we had a full hour for that one. That's really amazing. And we all have that weird uncle. So I am going to now pass it over to Ellen, who I don't believe needs any introduction.
Well, except I can't get my slides up. So let me just, Catherine, have you gotten out? Oh, they're up? Okay.
They're up.
Okay, good. So, you know, we've been talking a lot about privacy for the last 3 hours, and we've talked about this for a number of years. And of course, one of the questions we ask is, so why do we care about this? And we care about it because it may have some role to play in why people participate in research. It may have some role to play in how they respond, whether they seek getting medical information and how they respond to it. So building upon a lot of the work that we've already heard about from our group today, I'm going to share some lessons from eMERGE, which is the Electronic Medical Records and Genomics project, which has been going on now for more than a decade, as well as from GetPrecise and beyond. So let me start with one of the early things we did in our SEER, which is that we looked and saw what have people studied about what people think about privacy. And of course, it's a multifaceted project. But the fact of the matter is most researchers have spent most of their time asking about discrimination, and they spent most of their time talking about employment discrimination a lot. More recently, they've been talking some about government, but relatively little about the sort of normative notions about what people expect. Sort of interesting that that's what they've done. After that, in the second round of eMERGE, we also did a survey of 13,000 people who were patients at ResearchOne universities that are all involved in big league genomics research. And we asked them, we actually did a study to show whether they cared about broad consent or open consent or open access. Turns out it made no difference. Everybody approved, the approval rate was 70% across the board, just bang, that's what it was. But if you ask them specific questions, they had a lot of concerns. Almost half of them said that they would worry about their privacy. They would worry about their medical record being shared. They would worry about how researchers use their information. Interestingly, they're less worried about genetic information than they are about their medical record being shared. My favorite slide of myself is me on an airplane, and I'm more worried about losing my personal medical information than losing my DNA, period. But nonetheless, these are things that people said. Nonetheless, the overwhelming majority, at least hypothetically, said that they would participate. So then we did eMERGE 3, and this is a study where we enrolled about 2,500 people. We did broad-based genetic testing, and we returned results. And the citation there is the paper that we did all the information from Vanderbilt's site. There are actually a number of other institutions that are involved in this study, but their data aren't ready yet. But what we did was we compared the responses of people who got bad news, found out they had something, as opposed to those who didn't. And there were a couple of differences, but I want to point out some differences that weren't there. They tended to think that genetic information was somehow different from other medical information. In general, the group across the board didn't see much difference between the two. They were the people who got, quote, bad news were more willing to talk with their doctor. And needless to say, it had more of an emotional impact. That's no surprise. But what is really kind of intriguing here is that they were a little less confident about they tended to be a little less confident about their privacy, but not a lot, not statistically significantly so. They tended to have a little more privacy concern, but actually a little less privacy concern, but not a lot different. So it's actually kind of interesting that there wasn't much difference between the two. The other thing is that we did interviews with a lot of the patients who got, quote, bad news. And what they said was that they thought the results were helpful, but actually a frightening number of them didn't know what they meant and didn't plan to act on them. Now we're going to be getting the larger eMERGE group going forward. It'll be interesting to see what their results are, but that's interesting. So what are we to make of this? Because recruitment is looking very different from what happens when people actually get their results. Well, one is that I think we pay there's a lot of interest in society going on now about genetics and its impact. And you've heard from Jay about how much this is in literature and film and TV. And also, there's so much emphasis on this in research. The fact of the matter is, it seems to be just about the only ELSI question that people can ask. Another thing to say is that we are in an era now where we have pervasive public concern about privacy, about government, not only in forensics, but also surveillance in general, and the notion of big tech. There's also the history of distrust in research, to which Mark alluded very early on. And this is a large issue, and it falls differently across race and social class. And then I think what we see also here is some concern about whether the results, how valuable the results are in a research context where you're not going in with a specific question to be asked and answered. And I would say that, what I would say is that the focus now needs to be on, this is where the focus needs to be on trustworthiness and thinking about privacy. In return of results, the reason that looks so different is that these people have already chosen to enroll and they're enthusiastic about getting information. It's not surprising that they think it was a good choice because frankly, you have to feel good about the decisions that you made. I think the concern here is that they didn't do much in response to genetic testing. So I think that the need for continuing effort to protect data is great. It's interesting that you all talk about very sophisticated ways to attack data, but the fact of the matter is current practices seem to be work reasonably well, despite the possibility of attack. My hypothesis for this is that there are a lot of incentives not to do attacks, both legal and ethical. The legal system, however, is a sieve, and that frankly, the social context is becoming more fragile. So the price of, the price of this research is vigilance. Thank you very much.
Thank you. And we are on time, XiaoFeng, totally on time. So I believe that we have time for questions that we can now open up to the audience.
Certainly,
so
certainly. As I told you, you have 45 minutes. Now I think spend about 30 minutes, and I have another 15 minutes.
That's amazing. We pulled it off. All right, so I will open the floor.
Hi,
Hello, am I on? It's Carl Gunter, University of Illinois.
Okay, Carl, go ahead.
Yeah, this is a question for Ellen. I was wondering, or all of you, but which is, how do you think the things you've learned will relate to the All of Us program? Is it bearing out as you thought, or are you be able to feed any value into it?
Well, we certainly hope so. I think that the whole point of what we're trying to do here, and what I'm not quite so directly as I wanted to say, was we think the holistic approach that we're taking actually gives better insight into the lay of the land about how we ought to proceed. And I think that that in terms of all of us, what it really says is that the emphasis needs to be put on the front end, and to really think about creating transparency, trustworthy mechanisms, so that people can have some trust that that data about them are going to be protected. I guess I should ask Brad, but I think this is really important for how we conduct this research. And Catherine, I want to just say one thing about her. The issue that she talked about is just about the hottest issue, or the most contentious issue in genomics right now. Can we really talk about mental health? Can we talk about sexual orientation and gender identity? Can we do this research? And one of the things that everybody that Catherine talked to said, no one has ever asked us before. No one. that should be a clarion call. Sorry, I'm an advocate.
No, that's fine. Let me synthesize real quick across a lot of this with respect to all of us, since I'm so intimately involved in that program. I think one of the things that all of us has done, and I wouldn't say that this research specifically influenced it, but it's partially motivated by it, is that all of us has recognized that there are a lot of underrepresented groups in biomedical research and has made a concerted effort to overrepresent them within, or at least recruit on a much larger scale than what has been done in a lot of other cohort studies in the past. And so at the same time, though, we've been very careful about dissemination of information on populations or subgroups that that are, in some situations, are considered to be a little bit more sensitive. So for example, in the release of the data that we just put out about a month ago, we actually did not put out the information on LGBTQ+. This is going to be provided in the next tier of access that we make accessible, and it's been interesting because on one side we've had that population, that group, representatives from that group say that they want this data to be out into the public domain as fast as possible. At the same time, we've had pushback and said that the program itself feels as though it needs to get some legs underneath it before it starts putting all of the data out into the world. And so there's this process towards getting information out there. So I'll pause.
Thank you.
Did anybody else on the committee have something to say about that?
Yeah, hi, this is Prashant. Oh, sorry.
No, sorry.
I was going to ask a question, but go ahead.
We'll start taking a line though. Hold on. So I'm going to get Prashant and then I'm going to get JP. Okay, Prashant, go ahead.
Great, thank you. So thank you all for very interesting talks. I mean, one of the things that I was actually wondering is how much this conversation between scientists, geneticists, genomicists, and people on the ethical side of things, the legal aspects of it, people who have a stake in it, clearly like Ellen mentioned, like mental health, for example. So in terms of, I understand that the IRB process is meant to be sort of a gatekeeping process to a certain degree, but how much room is there, or how much do you see there's value in having at the granting stage itself, there be a panel for certain types of topics to be discussed with people from a broader range of expertises?
Ooh, you just threw the easy question out there, didn't you? Okay.
Prashant, let me, if you don't mind, the statute actually prevents the IRB, the regulations prevent the IRB from looking at the ultimate question, which is what the downstream consequences are going to be. And so I think that IRBs are actually, and frankly, given that all of us typically work with de-identified information or under the common rule, not even very de-identified, the IRB formally has no role other than to say that they don't have anything to say. Mm. And so, so the model that sort of emerged is one of a, I wish I didn't use that verb so frequently, but has come up, is sort of this idea of embedded ELSI. And the truth is, at Vanderbilt, I meet all the time with the geneticists, and all of us meet all the time with the geneticists. And so they're really very used to, they're really very used to reaching out and sort of having this conversation. And so I think that really, if there's going to be anything, I actually think that more effort on sort of being transparent about what you're doing, about reaching out to the community and asking them what they think, looking at some of the Jejun's data, about what people spontaneously say. I think that kind of responsiveness is actually more authentic and more probative than what an IRB does. Because they have a very specific, the function of an IRB is to protect the institution. That's its real function. And so, and I think that our project here is to protect real people. How about that? That's snarky. So, Jay, I want
to jump in with a really short answer, which is that your question makes me think of the contrast between the United States and the European Union. The European Union has specific guidelines that require the consideration of cultural and social consequences before they articulate policy. I'm thinking particularly in the area of GMO regulation.
Interesting. Just to play this out a little bit further, I think there's from a policy perspective, there's a little bit of a challenge, at least in the US, that we run into, and this was why I was pushing the issue earlier on GDPR and genomics, in that we still, at least under the common rule, it's pretty obvious that genomic data does not have a designation of human subjects. They've been very clear on that. And then HHS kind of has followed OHRP's lead in saying, well, we're not going to designate DNA as an explicit identifier. And so it kind of has put things into this little situation where organizations and individuals are left to decide for themselves how they want to manage that information. And so Vanderbilt, for instance, took a tack in that, like, we created a community advisory board that we get feedback in terms of the types of research that should be done, what types of protection should be put in place. But it's not a federal requirement necessarily, because that's not something that the law is affording them. So it has created a little bit of a cowboy country in the US as an artifact. And based on the discussion earlier today, it sounds like the EU might also be in a little bit of a strange situation. But so, like, what role does the IRB have? Sometimes it becomes, it's up to the institution at this point, because if it's not a human subject study, if you're dealing with the identified genomic data, then, if the institution wants to do diligence, then they'll go and they'll do the review. Otherwise, legally, they don't have to.
Thank you all.
Okay. He had his hand up.
Yeah, JP has been kindly waiting, and as I said, it's slowly turning into, what time is it? 2 in the morning now?
Come on, it's 10:08 p.m. right? It's still very humane, right?
I know.
Not exaggerating. Yeah, no, my question actually is somewhat aligned to the last comments made by yourself, Brad, and Jay, which is I think this is fabulous work. I think you should absolutely continue this endeavor. I was wondering how you generalize this further, beyond the US, and so you touched sometimes to the EU, but there is way more, right? I mean, after all, the US is something like 5% of the world population, and the EU is another 5%. So this leaves still out the 90%, right? Of course, your center is in a given location. This is understandable that you take this locality into account, but we're talking about a very global issue, right? So how do you generalize this to take into account, places such as, Brazil, China, Japan, Nigeria, that are, people have DNA there as well, as we know, but they're in completely different contexts, right? So did you give any thoughts to that aspect?
Let me jump in on that one first, if people don't mind. So one thing I will say is that when our center was first funded, we explicitly put the focus on the United States, given the highly given the uncertainty about some of these issues. So we were actually just refunded by the NIH about a month ago, and the whole second iteration of this is to be about the international perspective. So we're very much at the beginning of this. That said, what you're alluding to in terms of this broader multinational perspective is it is really challenging for a bunch of Americans to step in and look at the culture of other countries and try to interpret things from that perspective. And so it's going to require active collaboration. It's going to require finding people who want to work on these topics within their respective countries. And sometimes that's not simple. not just because you can't find people, but sometimes there are political issues that may prevent people from doing this type of investigation within the countries that they're in. So it's not a simple problem.
So what I would say is that the real strength of what we've done here is the methodology. I mean, the idea that we, you know, that, I mean, I'm a lawyer. And so in addition to being a pediatrician, and as is Catherine, and so I do a lot of just straight up legal stuff. And that's, you know, but the idea that we use multi-methods to try to understand the cultural context, ranging from, you know, what people are looking at on the television to what they're saying sua sponte and social media, to all of these other things, and history, which I didn't allude to earlier. I think that the idea that all of that has to come into play, you know, is a challenge. And I mean, and I was involved with the CIOM's genetics guidelines about 20 years ago. And I do realize that, you know, that it's hard to say anything, well, you can't say anything very universal, but I think what we can say is that taking a single perspective and thinking that that's going to solve the problem is a big error, I think. So that, I mean, that would be, that I think is what generalizes, is the idea of a broader scope of analysis.
I'd also like to just supplement this by saying, I think the world has changed rapidly in the last 4 or 5 years, and is going to continue to change rapidly in terms of commercial entities becoming involved in this entire process. The news of how DeepMind and Google interacted with NHS, and how now Verily and Google is now starting to do large studies in the US and is supporting part of the All of Us program. I think you're going to see some major changes around how things operate over the next couple of years, not just on, you know, locally, but also internationally, in terms of where data is allowed to fly, who's allowed to study it, and when. We haven't seen the big case come forward yet, but it will happen.
Thank you very much.
I think we are actually out of time. But we can keep going if XiaoFeng wants to give us more time.
Yeah, no problem, no problem. You know, I'm waiting for Justina to say no, but looks like she's allowing us to go forward. And you know, related to your comment, I think it's very interesting looking at the whole situation. It's not only the situation, you know, posed by political whatever. Also, there is also change in the underground business, right? In the criminal world. And so far we haven't seen any evidence that people using this genomic data and biomedical data for some really big illegal things. Of course, I'm talking about, you know, systematically, not as you know isolated events, right? But down the road, and with the sequencing become increasingly easy, and with the genomic data increasingly disseminated, and the underground, those cyber criminals may, you know, find a way to make use of it, to monetize it. So when this happens, and the policy will change. So my question is, are we gonna wait for these things to happen to do this reactive change, or are we gonna be a little bit more proactive, looking at what could be done, although hasn't been done yet? You know, just and do some proactive protection to ensure this kind of cyber criminal ecosystem will never, you know, just become a real big thing. It will endanger a lot of people's privacy.
Well, I mean, I'll just make an observation about the United States right now. Right now, we are in an absolutely non-functional government. That, I mean, you know, in fact, Congress is usually reactive as opposed to proactive. That's the nature of the law. And to the extent that and sometimes it can be proactive, like the passage of the Affordable Care Act. But I think what we have seen, at least on the national level, is clearly absolute paralysis. And I'm hoping like crazy that will change, but that's the case. On the other hand, we may see some action on the state level. I mean, you can look and see what's going on in California currently with the CCPA to see if that turns out to, I mean, we talk about the laboratory of the states here in the United States. You know, whether there are going to be some interesting, actions. But I think many of us would like our government to be more proactive, but it is in the nature of governments not to be more proactive. And to say nothing of what we've seen in the last 4 years, which has just been shocking. So, in my view, and not to be at all political, but as Jay is laughing. But there are bills pending in Congress. You know, nothing's going to happen until after the election. And once we know which way the country is going, I think we'll have some idea. So
I will echo but I will echo the sentiment of government being reactive instead of proactive. It's always very difficult to, as one person once said to me, unless you see the bodies floating in the water,
oh yeah,
it's very challenging to convince people that there's an actual threat. I mean, because until that threat is realized, it is nobody knows how close it actually is. And that doesn't mean that we shouldn't be proactive, but you know, at least on the American side, it is, you know, it is, I want to say very challenging. It's like beyond very challenging. It's like challenging times infinity to try to institute that type of change, because if it ain't broke, don't fix it. So, you know, when you have a catastrophic failure, things will change. But, you know, I'm gonna quote you, XiaoFeng. I think this was like 6 years ago. We were at a meeting and you said, you know, let's try a solution. And, you know, what could possibly go wrong?
You still remember that?
Of course I remember this, because what do you say? You said, if we get it wrong, what's the worst that happens? We just screw up privacy for all of a generation. And
yeah, but now I guess I changed my mind. But looking at, I'm not talking about privacy. I'm just looking at the current situation, right? And you know, back 6 months, you could never imagine, and we could stay home. You know, I've been stay home arrest for how long? I don't even remember. 6 months at least. and you never think about the things become so big. So I really think in every perspective, whenever the things could have a huge impact, we should be a little bit more proactive.
So let me make a suggestion here, if you don't mind. I mean, one of the things that you all have come up with a number of really interesting methods to try to protect privacy. And when you are thinking about it, or perhaps even writing about it, or you know, talking with each other, think about what possible policy solutions would need to be into place in order for those things to be implemented. I mean, you know, doing policy is no joke. I mean, it's really hard work. But you know, but if you think beyond, how can I solve this problem, and think, how can I implement this in a way that's really going to, you know, that's really going to get where we want to go? Because at the end of the day, this is about deciding where you want data to go, and how, you know, and how much control people ought to have, and under what circumstances. That's the question. And so that's where you're going. And so think to that next step. You know, you guys are the pros from Dover. Give us some ideas about what to do.
Yeah, wonderful. I think it is a very good suggestion. And Brad, it looks like Heidi have something to say.
Well, Heidi always has a lot to say. I was hoping she would say it instead of typing it.
Oh, what you said is, are we going to look at international data flow? Of course we are. How can we
No,
not?
no. Heidi's last comment is about how,
have you turned her on?
Do I have to turn her
on? She is not a panelist. I guess so.
Hold on. I don't know if she's still here.
Yeah, I am. So, Brad, I'm actually quite encouraged by your comment of how hard it is to kind of get the effect of people not really wanting to deal with it if they don't feel like they have to. It's actually kind of encouraging to me, because, you know, it is pretty remarkable given maybe the nature of the potential threats. But I guess, so it seems like one approach that companies like Big ID are taking, and other companies, is that you're not just I hate to use the word, but selling privacy protection, right? You're selling a kind of a broader data management package that has other, very tangible value to the companies that might purchase it. So, with a corporate takeover, company comes in, they have real trouble just knowing what their data assets are. That's a huge challenge. So you can offer a package that allows them to know what they have, what they've acquired, where there's redundancies, how it's structured. And then also, part of that is, it's the first step to actually providing privacy protection, because then you understand the different sensitivities and different liabilities, different kind of potential legal exposures. So it's not so purely on its own. You have to kind of make an argument for a catastrophe, but there's a broader context where the same types of strategies can give you privacy protection plus all these other benefits in create a kind of a broader argument for why somebody should be interested in it. So seems to be springing up, but Big ID is the company that I've seen do this.
I would say I don't think that making the argument through catastrophe and trying to avert a catastrophe from with respect to data is really going to get very far. You have to kind of, you always have two options when you're defining policy. One is you can use the stick approach, which is that, I'm going to tell you that if you do something wrong, or if you go beyond the boundary conditions of what you're supposed to do, I'm going to whack you with the stick and you're going to get back in line. The other one is the carrot approach, where, you give people incentives for doing this. And in this situation, what would the incentives be? The incentives would be twofold. One would be you can support things that you would not have been able to do otherwise. Now, we've seen clear examples of this throughout the day with respect to how thinking works in the EU, and that like data sharing, like this notion of centralization, like what we've done in the US, you do not see that in the EU. And the only way that they could get to some of these types of analytics would be to have these technologies at the table, right? We don't necessarily have that same incentive structure in the US. But the other way to do it would be this perspective of almost insurance. I hate to sell it this way, but in the event that something was to go wrong, right? Where is liability and how do you manage it, right? And if you have privacy protection technologies, better security in place, maybe you can mitigate the risk and lower the cost of the violation that takes place as an artifact. But it's difficult to just sell this as part of a management schema, because at the end of the day, privacy and security will probably always be thought of last. People will say that they want it, but but service comes first. I think everything we've seen is that you offer somebody a great service, or you create that new crazy service. It's privacy and security comes after the fact, and whoever comes into it and uses the technology, they're there. So now maybe that's a bit of a bleak and hopeless way of looking at the world, and it's probably more than just two options of carrots and sticks. But it's that I will stand by that for the moment.
Yeah, I just want to briefly touch on something that came up earlier, which is, the idea of well, kind of the more theoretical understanding, or the full, full-fledged technology and research aspect of investigating privacy. But then when you get down to application, being able to really understand your attack surface, I think is really important. And that's a whole separate research domain on its own. And I would love to see those two things come together, where there's, an understanding, of the attack surface, which means you have to actually be honest about it, because a lot of the biomedical researchers, they just, like you're kind of alluding to, they just want to be in their happy place, and they don't want to have to worry about it. They don't want to, they want to imagine that the problem doesn't exist. And that's, I understand that viewpoint, but if you're honest about the risk, then you say two things. You say, well, you can't just do that. You can't pretend the risk doesn't exist. But then the other thing is that you don't pretend that every possible conceivable threat is actually something that needs to be addressed. It's got research interest, But in terms of pragmatic application, it's those two viewpoints coming together that maybe are most interesting.
XiaoFeng, if I can say one word.
Yeah, go ahead.
So
Go ahead.
to add to what Heidi had said, I'd like to bring up a very simple application app, WhatsApp. So the whole success is based on the fact that it's almost as good. It's as good as alternatives, but it's claimed to provide you security. The moment a good alternative, like the one Ko had presented, where the privacy comes at very limited cost, and the outcome is as powerful, you don't have to have a very big stick. The moment there is a threat of a stick, people may just switch. So I'm not sure I'm as pessimistic as I was a couple of years ago.
If I may add one more thing. So yeah, I would like to add to what Zheng is saying, and to say that it's not only the matter of making things run faster, it's also the matter of infrastructure. Because, and this is what scholars in science technology studies say about infrastructure, right? That when it fades into the background, it becomes invisible. So when security infrastructure, something like public key infrastructure, something like symmetric key encryption, or SSL, something like that, when it fades into the background, it becomes very difficult to challenge. Now, this can go different ways. If it's an insecure infrastructure and it fades into the background, it's really difficult to dig that out and turn that into a conversation, which is something that we have issues with launching new technology. But if we are willing to work with different frameworks, mainly legal framework, political framework, to turn things into infrastructure, that would be where things can be effectively implemented. So, getting some interruption. So something like using SGX as a default without making that a question, that will be something that's really powerful, even though it doesn't protect against everything, but it's an order of magnitude better. So I think we need to think more about how public infrastructure plays into how we can engage with security.
I would second that, and I would say that, but we still run into challenges of setting defaults. And so, for instance, if you look at HIPAA, encryption is not by default. Encryption isn't addressable. It is not required, which is absolutely astounding when you think about it. The fact that SGX, that most modern most modern systems today, most modern servers and phones have the ability to do SGX it boggles my mind that it's not used more. But it's there, and yet, you know, the infrastructure, I wouldn't say it's default, but it's there, and we still don't see enough applications of it. I don't know, that's all I'm gonna say.
Yeah, if I may just have one last sentence. I think, yeah, that's a really great question. And I think it has to do with the process of standardization, which is more complicated than we think, how cryptography becomes standards. So that's one subject to add to transdisciplinary research, is to study how exactly technology becomes standards and becomes infrastructure. It's not something that's obvious at all. And it's a really messy and complicated process. Thank you.
Oh, if you want to know, just try becoming part of an ISO committee and just watch things play out over the course of a decade before like anybody agrees on what a single word means. That's frustrating.
Yeah, now it's 2:30. I guess we need to worry a little bit about the people in Europe, right? so you know, Brad, do you have anything to wrap up your panel? I think it's wonderful.
I think we're good. I think that we've said all there is that we need to say.
Okay, great. Thank you for all you guys for such a wonderful, inspirational panel. You know, we're running half an hour later, but still we have more than 50 people in there. You know, just I think 54. and in the workshop. I think the attendance is great, and thank you for you all for the just enjoying the board 3 hours and a half. We're supposed to be 3 hours, but 3 hours and a half. Everyone feel happy, yeah. And I think this is the best time I spent, I think, in this week. At least for this week.
XiaoFeng, the week just started.
Yeah.
We
okay, great. So yeah, and I think I never thought we can be so successful and for an online workshop. So, but I'm a little bit worried because next time, if I prefer, I still want to travel to Australia and talk to you guys face to face. Also enjoying the beautiful thing outside and whatever. So, and I think JP is gonna say something about the next workshop and ask the steering committee chair.
Okay. Well, XiaoFeng, so let me first second you in, let's say, thanking all the contributors and all the speakers. I think these were indeed very enjoyable 3 hours and a half. And actually, I think you made the best out of this, let's say, available channel. So I think it was very pleasurable to follow and to see how actually challenging 7 years later the topic still is, right? So we're really touching something that is really very, very crucial. And we see more interest in this workshop, so we should certainly continue. This is actually the 3rd time we collocate with the GFOGH, right? Well, collocate, of course, in today's world doesn't mean that much, but still we are, let's say, co-organized with the GFOGH, and so in some way we, let's say, we contribute to their visibility, but they also help us in making the GenoPri' workshop more successful. So I think we'll try to continue this this collaboration in the future, and we will see where we are in 2021. But certainly we'll expect GenoPri' 2021 to take place sometime in the fall. So thanks a lot, and hope to see you soon. Thank you very much. Take care.
Thank you, and I hope next time and next year we can see each other face to face, and we can have a lunch together, dinner together, instead of doing these things remotely. I sincerely hope this nightmare just gone quickly.
Good.
Okay, see you.
See you guys.
Thanks a lot. Take care. Yeah.